Good morning to many of our colleagues who are joining from abroad, and of course, good evening to our Indian colleagues. It is indeed a great pleasure for all of us to have you with us, of course, virtually. But it is a tremendous opportunity for me and also all my staff and others who are going to participate on this very important webinar on genomic strategies for improvement of abiotic stress tolerance in croplands, which is being organized jointly by ICR, National Institute of Abiotic Stress Management, which is located in Baramati, Pune district of Maharashtra, India, and of course in collaboration with the Alliance of Biodiversity International and International Center for Tropical Agriculture, CIAT, in its Asia India office, New Delhi. I welcome each and every one of you. Our moderator, Professor Bansal, who is known to all of us, all our eminent speakers of the day, and all the participants. Friends, let me welcome all of you with our institute song. I think you will enjoy the song.
खोज खोज पर तनाव मुक्ति का नारा है, संस्थान हमारा है, संस्थान हमारा है। यहाँ सोच नहीं पर त्याग वही, किसान को प्यार की वही, उन्नत खेती की सब प्यास बढ़ी, याद लिया धमकी हुई खड़ी, कृषि परिषद का विश्वास है, विज्ञान जगत का कौशल भी बारामती पेड़ से लाभ में सहारा है, जैविक स्थित प्रबंधन संस्थान हमारा है। क्रांति के गहरे रंग खिले सुरक्षा है लक्ष्य यही, पशु पक्षी मत्स्य उत्पादन बागवानी से आय दुगनी, हवा पानी मिट्टी से यहाँ समस्त तनाव मिटाना है, संकल्प सिद्धि का झंडा गौरव से लहराना है, जैविक स्ट्रेस प्रबंधन संस्थान हमारा है, जैविक स्ट्रेस प्रबंधन संस्थान हमारा है।
Thank you very much, friends. As you all know that abiotic stresses affect agriculture. And the stresses, it could be moisture stress because of drought or flood, nutrient stress, whether it is high or deficiency, radiation, temperature, pollution, salinity, mechanical, all of these abiotic stresses put tremendous pressure on growth, productivity, and also profitability of agriculture. And unfortunately, all the stresses are intensifying. As you all know that climate change is aggravating, water stress is increasing day by day, soil quality is declining, pollution is also accelerating, and along with all these abiotic stresses, several biotic stresses are also invading agriculture. And you also know that the frequency as well as intensity of all these stresses are going to intensify, whether it is cyclone or heavy rain or heat wave, flood, drought. IPCC, that is Intergovernmental Panel on Climate Change, and also our own assessment shows that in years to come, all of these stresses are going to be intensifying and becoming more and more fiercer on this. But fortunately, genomics tool can really help us. It can really help us in mitigating and also adapting to several abiotic stresses. Whether we can call it from gene mapping, gene sequencing, gene identification and functional elucidation, DNA marker-assisted selection, or molecular breeding, CRISPR-Cas, GWAS, and you can name several of the emerging technologies which could be of immense use for managing, for addressing the stresses of abiotic. But there are still several challenges. The challenges include, there are multiple stresses which affect cropland. It is not only one stress, there are multiple, and many of the stresses work simultaneously on one individual plant. Second one is the molecular mechanisms for response and also resilience to stress are not fully understood yet. There are several progress, lots of progress, but still it is not fully understood. Thirdly, that large number of plant genetic resources are available, but only a few are characterized for resilience. So still there is a long way to go. And of course, only a few genes functionally elucidated for their role in multiple stress tolerance. That is another aspect. And again, when we move from greenhouse to the field condition, another multitude of stresses start affecting the crop plant. So all of these challenges even today persist. But even though there are several opportunities, as I was mentioning, that there is significant advances taken place in genomic research for improving abiotic stress tolerance worldwide in several crop plants. There are several success stories available, and those success stories can serve as our milestone. And today, again, we will be listening some of those success stories. Indian science, if we call it within our country, has also made tremendous progress, significant progress in abiotic stress research. And of course, global collaborations are available to intensify the efforts. In today's endeavor, today's webinar is going to explore several of those opportunities where we can really start collaborating for abiotic stress research. Our institute, that is National Institute of Abiotic Stress Management, as I was mentioning, located in Pune, Maharashtra, is working on this aspect. And our mission is managing abiotic stresses for sustainable agriculture. And we address this particular mission through 4 of our schools. School of Atmospheric Stress Management, Water Stress Management, Soil Stress Management, and of course, Social Science and Policy Support. In several of these, we will see that we really work on the genomic aspect of atmospheric stress management, whether it is water stress, so abiotic stress management. And today's webinar is one of the several steps which we have taken to achieve our mission. So the title of today's webinar is Genomics Strategies for Improvement of Abiotic Stress Tolerance in Crop Plants. And our specific objectives of today's webinar are: number 1, understanding the mechanisms of abiotic stress tolerance in crop. Secondly, developing genomic strategies for improving abiotic stress tolerance. And thirdly, exploring opportunities for collaborations for abiotic stress research. And all of you will agree, my friends, that you simply cannot have better speakers than we have got today. We have got Professor Mark Testa today from King Abdul University of Science and Technology. We have got Dr. Hima Bindu from ICRISAT, Hyderabad. We have got Dr. Matthew Reynolds from CIMMYT, Mexico. We have got Dr. Girder Pandey from South Campus of University of Delhi. And of course, we have got Dr. Bishanathan Chinnaswamy from Indian Agriculture Research Institute, New Delhi, my previous organization. And to organize and to moderate the whole thing, again, all of you are very much familiar with our own Professor K.C. Bansal. Dr. Bansal doesn't need any kind of introduction. All of you know him very well, and currently he's associated with the Alliance of Biodiversity International and CIAT Asia, India. And of course, our convener, Dr. Ajay Kumar Singh, he has also put a lot of efforts, tremendous efforts in organizing, putting everything today for this particular webinar. Friends, with this brief introduction, I welcome all of you once again virtually to our institute, and I wish that this particular webinar is really going to enrich our knowledge in terms of genomic tools, and we will be really learning several new things. We'll be able to explore the new opportunities of collaboration. With this, once again, I welcome each and every one of you, particularly our speakers, our moderator, convener, and all of you. Thank you very much. And now I request Professor Bansal to please take it forward. Professor Bansal, the screen is yours, please.
Thank you. Thank you, Dr. Himanshu Pathak. I think you have already covered several aspects regarding the webinar, but again, on my own behalf, I have a pleasure in welcoming all the participants and the eminent speakers, actually on behalf of the Alliance of the Biodiversity International and the International Center of Tropical Agriculture. And I'm particularly thankful to all the eminent speakers, in fact, who have accepted our invitation and also for sparing their valuable time. Though Dr. Himanshu Pathak has already covered a few of the aspects of today's webinar, but still, I think in few more minutes, I would just like to take along the young colleagues and others who are joining us today for this webinar, that increasing crop productivity, particularly the rate of crop productivity and the total production, is always an important agenda of research for any international or national institution. And compared to the 20th century, in 21st century, the agenda has rather become more challenging because we need to achieve all that high production, keeping in view the sustainable developmental goals of the United Nations. So therefore, it is important today in this era of climate change that we develop or breed varieties which are tolerant to different abiotic stresses or to multiple of these stresses. While plant breeders, no doubt, have developed very elite genotypes, high yielding, resistant to various diseases and pests, but then breeding these varieties, for different abiotic stresses is a very challenging task. Simply because, that these different abiotic stresses, as we hear more from the experts, are of very complex in nature, and different mechanisms we have deciphered so far also turned out to be quite complex in nature. And be it, we talk of drought or salinity or high temperature stress or even nutrient deficiency stress. They're all of, in fact, really speaking, quite complex in nature. So therefore, genomic strategies, which have become available in the last about 25 years or so, have certainly helped us and has been able to identify or decipher, elucidating many of these, molecular or genetic mechanisms through the identification of signal transduction pathways, or some of these, complexity could be deciphered further so that we can identify some of those genes or genomic regions for use in, either genetic engineering or molecular breeding or genomic selection, or today we talk of genome editing. But again, as physiologists, we have been struggling quite a lot. In fact, in giving some kind of a physiological selection criteria to the breeders, but not a single parameter you could ever use, really speaking, under different target environment conditions for breeding of these, abiotic stress tolerant crops. But then now we have genomics and also we have phenomics based on high throughput, phenotyping. So therefore, combining these efforts of phenomics and genomics, there is a possibility that we'll be able to sooner than later probably develop these genotypes which are adapted to various climatic conditions and different abiotic stresses. And we look forward to more of smart or climate resilient agriculture. And in fact, also it has been analyzed internationally that any enhancement in yield overall, because we have not only the seed, we have all those cultural management practices, particularly for these abiotic stresses. But then it has been analyzed that 70% of the total increase in production out of 100% productivity is going to be attributed to the genetic gains. So therefore, it is important to study the genomic strategies for improvement of crop plants, not only with regard to abiotic stresses, but in general. But however, as I mentioned, and as also Dr. Himanshu Pathak mentioned clearly, that it's more of challenging task than, breeding for some of the irrigated or environments or optimal conditions. Breeding for suboptimal conditions, with weather events which are quite variable, and dwindling genetic or dwindling all these biological or natural resources is rather more important. So with this actually, back in mind, this webinar has been organized, and fortunately, as you see, we have 5 eminent speakers who are experts in their own fields, and thankful to them that they kindly agreed to spare their valuable time. And as per the program, as you know, total we have 5 speakers, and the first speaker who is going to give a lead talk is Professor Mark Tester. And I have a pleasure in introducing very briefly, though Professor Mark Tester to this audience needs no introduction, but still I think I'll take a few minutes to introduce him to the audience, to the participants, that Professor Mark Tester is a professor of plant sciences and also the associate director of the Center of Desert Agriculture at the King Abdullah University of Science and Technology, Saudi Arabia, since 2013. And before 2013, he was a professor at the University of Adelaide and the Australian Center for Plant Functional Genomics. Professor Mark Tester is a world-renowned scientist. He has contributed significantly in deciphering the molecular genetic mechanisms for developing crops, tolerant to particularly high salinity and also high temperature. And of course, he has several, publications to his credit, high impact journals. And I happened to see one of his reviews, written only about 10 to 12 years ago, has already been cited about 10,000 times, one single article. So we have a pleasure, dear participants, that one of the leading scientists of the world in this area of research is here today to give his talk. And I have a pleasure inviting Professor Mark Tester for his presentation, please. Professor Mark Tester, I request you and welcome you and kindly join us for giving your talk. Screen is yours. Professor Mark Tester, please.
Thank you very much for your very kind introductions, very informative and helpful introductions as well. So I provide a very good context. Thank you very much for the invitation. It's an honor to be here and to be talking in such a good institution as part of this good national organization, top national organization in one of the more important countries in the world. So thank you very much. It's a pleasure to be here. I'm going to share my screen now. I have a PowerPoint presentation. And you must tell me, you must keep me under control, Chairman, and cut in when I'm, if I'm going too long. So I gave a title of a talk which I thought matched the theme of the conference. But as I was preparing this talk, I actually thought I might slightly do a slight nuance of this title. But I am going to, of course, try to demonstrate some of the utilities of functional genomics, and in the introductions, I think very good to point out the phenotyping that we're also able to do, which I think the coming together, phenotyping and genotyping, gives us really exciting opportunities for doing big things with crop improvement over these coming years. So the context, I think, for many of us, and you're on the edge of the Deccan Plateau, and there's a lot of challenges in these areas for agriculture, for sustainable agriculture in particular, where you've got limiting rainfall, is that the context for our work, especially in the Middle East, which I do think has significant relevance to areas like in India, elsewhere in South Asia, is that a lot of our food does come from irrigation. And by far, most of the water that us humans use is for that irrigation, for agriculture. And this is leading to really significant challenges, which actually worry me, I have to say. We see that every aquifer that is being used for irrigation, and that contributes, getting on to half of all the water required for irrigation, and every aquifer that is being used for irrigation is being depleted. And this is the corollary of this is that these irrigations methods in these systems, which is contributing a third of the world's food, is not sustainable. We have to change. And of course, there are many, many ways that we can change. And we need to be thinking of several approaches. And of course, one of these is to try to reduce our use of water. And another approach is to see if we can start using water that we cannot currently access. So we're only using a tiny fraction of all the water that is on the planet. Less than 1% of the water that's available on the planet is used by humans. Of course, most of the water is in the sea, and it actually has many useful nutrients. It's got lots of good properties, seawater. It just, in inverted commas, just has high concentrations of salt, which of course provides us with a very important fundamental challenge. But this is a challenge which in research institutes and in universities, we should be rising to and trying to do things that aren't of instant reward, but have a longer term payback. And for me, over the last decade, building on my previous research, I've been really wanting to see if we can develop technologies, approaches to unlock seawater and unlock other water that we currently cannot use to try to contribute to global food security. And if we're going to unlock something, we need a key. And for me, now, as was stated really well in the introductions, I think we have a couple of keys in our hands now. And those keys include genomics, which is turbocharging genetics. And so it's really giving us the opportunity for real revolutionary change. And that's very, very positive aspect. So just as medicine is facing a revolution powered by genomics, so too is agriculture, because of this turbocharging, if you like, of genetics and ultimately breeding. And we also have huge opportunities with phenotyping. And so together, I think these do provide us with very significant opportunities for serious innovation. In particular, I think it gives us opportunities to unlock wild germplasm and to do things that really weren't possible, very practically possible for a long time. So we want to unlock wild germplasm and do two things. We want to get existing crops that we're already growing and using and try to increase their salinity tolerance. And the other thing we want to do is try to get plants which are already highly salt tolerant and domesticate them, change them to become more like crops, to have increased utility for us humans. So on the right-hand side, I've got an example where we've got salt-tolerant wild tomatoes, and we're trying to, actually do several things. One of the things we're trying to do is introgress the salinity tolerance from the wild tomatoes into the domestic tomatoes through fairly traditional, if marker-assisted selection breeding programs. And yes, there are other approaches as well, which we can talk about later if you would like. But then just for the purposes of this slide, the mirror image approach is to get plants which are already very salt tolerant and try to make them more like crops, increase their domestication. This is a crop which is already partially domesticated, but has really very high salinity tolerance. It's quinoa. And this is growing right on the shores of the world's biggest salt lake up in the Altiplano of Bolivia, Salar de Uyuni. So this is an example of work that we published quite a long time ago now, where we were taking this first approach, getting a plant which is really pretty salt sensitive, durum wheat, a tetraploid wheat, there was actually a wild relative which had salinity tolerance of some greater degree because it was able to exclude sodium more efficiently. And we actually identified the gene that was in the wild relative, introgress it to generate near isogenic lines and do field trials. Now this plant, the original plant, was identified by Rana Munns, a very dear friend and fantastic scientist and great collaborator. And she really was the primary driver for this work. And I was lucky to be a close collaborator with her. And when these plants have been grown out in the fields, you had fields with different levels of salinity tolerance. What we found was that when the plants have been grown in areas with low salinity, there was no yield penalty in the lines that had the gene relative to their near isogenic relatives that lacked the gene. So that was really good result. It wasn't bad to have that gene on board in the plant. And then when we grew the plants in the very high salt parts of the field, we got a significant, statistically significant, quite substantial rise in yield. So that's a nice result. And the science was good and we knew exactly the mechanism. It was a sodium transporter that was retrieving sodium from the transpiration stream in cells surrounding the xylem to reduce the amount of sodium being delivered to the shoots. The problem with this work is that although we did go out and do field trials, we tried to make the work as relevant as possible. Somebody should just That's better, yes, thank you. What happens was that the gene was really only giving a yield benefit when the yield was being greatly reduced in the first place by the salinity. So to give numbers to these, because this is the percentage effect of the gene, down here we're talking about yields of 3 tons, 2 tons, 1.5 tons per hectare, these sorts of yields. This is in dryland agriculture in, not fantastic conditions in Australia. And then this big yield increase was only when the plants were producing 1 ton per hectare. The plants were severely inhibited in growth. So good science, good result on the face of it, but the practical implications of this result, I think, are really sadly limited. So that was one example of where we've been trying to improve salinity tolerance. And if we're being honest, the true global impact of this benefit is modest, which is a pity. Okay, so when I moved to KAUST, I started to think a bit more and think about some other traits. That trait, I should have said, this trait was sodium exclusion. So when the gene was in the plants had about 1/5 of the sodium accumulation in the shoot. So intuitively, one might think this was a really good trait and would lead to strong yield benefits. But the strong yield benefits were only in very high salt conditions, at least for durum wheat. It may be better, this approach may be better in other crops. So I'm not being negative on the approach, but just a little bit cautious about getting too excited about the benefits in this particular species. But when I went to KAUST, I started to think and started to consider other traits which might be also contributing, perhaps more significantly than sodium exclusion, to salinity tolerance. And they were traits which I wanted to be able to measure at high throughput using automated phenotyping facilities. In this case, the plant accelerator that I built in Australia. It's a nice high throughput phenotyping facility. And in that facility, we're able to grow large numbers of genotypes, in this case of rice, and we're able to grow them in flooded conditions using a double pot system, and we're able to grow them in low and high salinity. These plants had been genotyped by Susan McCouch and her collaborators at a pretty high density back then. So there was about 700,000 SNPs, which gives you pretty good statistical power and genetic resolution, especially in rice, which has got quite a small linkage disequilibrium. And we were able, this was done by a PhD student of mine, Nadia Al-Tamimi, one of my many Saudi female PhD students I have, and very strong result. What the trait that she was measuring, I should say, sorry, was the ability of plants to maintain transpiration use efficiency in saline conditions relative to non-saline conditions. Now, one might think that that's a fairly multigenic trait, and I'm sure that transpiration use efficiency itself is a highly multigenic trait. Lots of different things will affect transpiration use efficiency. But with this facility and with these numbers of genotypes, we were able to measure not just transpiration use efficiency, but the transpiration use efficiency in salt-stressed and not salt-stressed plants. So we're able to measure changes in the transpiration use efficiency in those several hundred genotypes and do the genetic analyses. And when we were looking at the indica varieties, because of course there's very strong population structure in rice, but when we were looking at the indica varieties, we found this very strong genetic signal for the ability of the plants to maintain their transpiration use efficiency at high levels in saline conditions relative to non-saline conditions. And the gene which appears to be responsible for this is annotated as a protein kinase of unknown function. Now, and this work was published a couple of years ago now, but we are now actually zooming in and looking in detail at that gene, and hopefully I'll be able to report more over the coming years. Is maintenance of transpiration use efficiency relevant in the field? Unfortunately, I can't give you any definitive answers on this. To be honest, we don't have the genetic and field resources for growing rice. We've had some excellent collaboration with Africa Rice, with a gentleman called Babu Kamali. And we do find that at least harvest index in the rice is positively correlated with transport. But I think we need to do more before we're able to make more any conclusions about the true relevance of this trait. But it's still some nice genomics-driven genetics for the moment. Okay, now, what I wanted to do, as I say, I was thinking about this work, and those are two examples of, I think, the primary theme, primary thinking in this meeting. But I wanted to throw something else out to the audience. You're a sophisticated audience, and you know a lot about plants and plant function. And I just want to share with you some of our more recent work that's taking a slightly different approach to trying to improve salinity tolerance of crops. I think it's driven partly by some of the factors that were mentioned in the introduction, I think by Professor Bansal. And you were talking about no, it wasn't Professor Bansal, I apologize. It was the director of your institute. I'm sorry. But it was a very good introduction. And he was talking about some of the real complexities of abiotic stress tolerance and how we still don't know a lot. And, you know, even when we've done work where we know a lot about, you know, one or two particular traits and we take it all the way through, we're still really not making huge impacts, huge inroads into improving the abiotic stress tolerance of these plants. And so I've been starting to think about the profitability, if you like, for want of a better word, of taking a mirror image approach, and that's getting salt-tolerant plants that are seriously salt-tolerant, and see if we can domesticate them, improve their utility for humans. A lot of our work is with samphires now, Salicornia, and other extremely salt-tolerant plants, including some very salt-tolerant relatives of tomato. But what I'll do today is show you the work which is most advanced, which is with quinoa, which is already partially domesticated, but for which we really want to understand more about the genetics of domestication and see if we can really improve the crop so it can become maybe not a new wheat for the world, but a plant which can be grown in mechanized ways efficiently in broad acre environments that are marginal in character. Okay, so yes, this is just a summary slide from one of my students showing that we're messing around way down here on the graph of salinity tolerance, but there's all sorts of plants out here which are highly saline tolerant. Let's understand their salinity tolerance and see if we can domesticate them. And as I say, a lot of our work at the moment is focused on not just Salicornia, but that group of plants that can grow in full seawater, unequivocally grow in full seawater, and which have many properties that are very useful. But let's stick to quinoa for most of today. This is a crop that you find in marginal lands, in seriously marginal lands, right up next to the shoreline of this massive salt lake in central Bolivia. It can grow in areas as a dry land crop where the yields are unbelievably low. And when this gentleman, the head of this particular commune in central Bolivia, when I asked him, what are you doing? This is really not a very impressive looking yield. He says, we have nothing else but quinoa. It's the only plant that they're able to grow. And they can grow enough for their families at least, and for some cash as well. So quinoa is a crop of marginal lands, but it also is a crop with massive potential. And when we grow this, for example, in the Tibetan Plateau in southwestern China, you can get enormous yields and incredibly impressive heads of plants. And it's also, of course, is fantastic for women and children because it's got high nutritional value, high protein, but also high in micronutrients, which is in fact paucity of micronutrients is the world's biggest health problem and a massive challenge in South Asia in particular, but also in South America. So we have lots of potential for quinoa. What we want to do is understand the control of growth and yield in quinoa, as well as, of course, the salinity tolerance, and then see if we can improve the crop. We want to target, of course, traits which will facilitate mechanized planting and harvesting, giving traits that give larger grains, higher yield, maintaining quality, and improving yield stability. I mean, the plant really does have a very large G by E, and it really does have a lot of problems, agronomic problems. We need to improve early vigor, dwarfing, low branching. A lot of these things you would say are really pretty much domestication traits or domestication-like traits. And it is a plant with high salinity tolerance. It can grow in hard seawater pretty well. So to start this, we wanted to provide some of the foundations for having a functional genomics approach to this. And so we sequenced the genome of quinoa, and that's fine. We've actually greatly improved the sequence since the original paper that was published 3 years ago now. And it's publicly available now, version 2 of the quinoa genome. So if any of you are interested in working with quinoa, do please get in touch with me, because we have much better genome sequence now. But importantly, for genetic purposes, we've also now resequenced it's over 1,000 lines of quinoa. And this is providing a very useful genetic resource. So we're now able to grow these plants in field trials in various locations, phenotype them in various ways, in particular using remote sensing approaches, but also, out there in the field with a ruler as well. And we're now growing these core collections across as many different environments as possible with germplasm that we have already now resequenced. And of course, making this knowledge publicly available, I think, is very important. So we have a core collection in particular of 400 lines selected across the genomic, the population structure, and they're now being grown in about a dozen different locations around the world. In particular, we've got very intensive phenotyping of plants being grown in 2 locations in China, in northern Australia, in the tropical location there, and in northern Germany. Also here, of course, in Saudi Arabia. So we're now, yes, the numbers vary depending on the field site and the conditions and seed availability, all the usual practical constraints. But it's, we really are focusing in particular on a panel of about 400 lines. We're trying to standardize phenotyping globally, and they're trying to reach a global consensus for phenotyping of these plants. These are cards, field cards that we can take out to try to standardize, at least to as best as we are able, the phenotyping. And then we can start doing some analyses. So this is, for example, looking at height as a pretty important trait. The plants are very prone to lodging in a range of conditions, especially in long days. The plant is a facultative short day plants. They flower earlier, the plants are shorter when grown in short days. And there is a relationship between the phenotyping in a growth cabinet for short day traits and the, if you like, the ratio of heights in short day conditions versus long day conditions. But of course, there's more than just a day length that's determining height. There's many other genetic factors as well. So, and this is just to introduce you some of the other phenotyping and how really phenotyping is starting to get very, very sophisticated now because of machine learning and collaborating with engineers and computer scientists, computer vision people is really, really important. And they start to do some pretty sophisticated things from images that we're able to capture from drones. I'm afraid I don't have time to go through this in detail, I apologize, but we are able remotely to be able to quantify plant height and a range of different traits. You end up actually with huge amounts of data, And so you have to use machine learning to try to identify the most important, what we call predictor variables. And then, so we actually, my research has, it's been an interesting journey. Back in Cambridge where I started, you'd sit there and say, oh, I think I can guess what genes are important. And so you take a reverse genetic approach, forget it. So then I go, no, we've got to listen to the plants. We've got to take a forward genetic approach. So let's look at, for example, sodium exclusion. So then we do the work with sodium exclusion, find the genes and try to, predict or tell the plants which traits are important. And we've really come now to a third stage in our intellectual journey, which I think is practically important, where I'm even giving up now trying to guess what traits are important for a lot of the time, especially for abiotic stress tolerance. What I'm saying now is let's just let the plants tell us what traits are important and use the machine learning, let the computer, tell us what traits are important by using machine learning algorithms to predict or to determine the most important predictor variables, predicting, for example, maintenance of yield in saline conditions relative to non-saline conditions. So now I'm not only letting the plants tell me what genes are important, I'm letting the computer tell me which traits are important. We end up with huge amounts of data, of course, now when you're using these automated methods of phenotyping plants. So this is another PhD student, another Saudi woman in my laboratory. She ended up with huge amounts of traits very, very easily. And what we were really having quite a lot of trouble, not only doing machine learning, but managing the traits as well. And all the students in my lab ganged together because they're all coming up with the same problems and coordinated, inspired, if you like, by Magda, one of my postdocs, who's now actually a faculty member at Cornell. They developed this app. And I really commend this to the students, the people actually gathering data in the audience. It's called MV App. It was published in Plant Physiology last year. It's fairly recently. And you can find it very easy. Just Google MV App, MVAPP. And it's a really nice app that helps you pre-process and manage and analyze your data. So then what you do is you can phenotype now on large numbers of traits in all sorts of ways, in all sorts of conditions, and then you can genotype the data now, and we try to bring these together for genome-wide association studies. And a lot of these analyses, of course, are in process. I'm going to skip over this. This is just describing the new genome, which is very, very solid now. And so I say, please get in touch with me if you want that. We have population structure in quinoa, which is very, very strong, with at least 3 very clearly distinctive groups, and in fact, arguably even more subgroups within the highlands. But this is quite interesting. I actually think quinoa was domesticated twice, once in the lowland and once in the highland. We're just now sequencing a couple of hundred accessions of some of the diploid and tetraploid, actually mainly tetraploid, near relatives, to try to nail this down a bit more. But anyway, you can start to do GWAS. So here, these are quite exciting results because you can find from different locations exactly the same locus, the same gene being identified. So here we've got on chromosome B7, there are 2 subgenomes in quinoa, so allotetraploid, with controlling plant height. And this is in Australia, this is in northern Germany. And we find that under there is just a handful of genes. One of them is a gene involved in regulating light control in development, which is pretty exciting. That's my favorite one. And then there's a couple of other genes. There's a protein RNA demethylase, which is also involved in the literature, according to the literature, in flowering time regulation. And there's also a ubiquitin protein ligase. I think there's a couple more genes, but those are just those are the 3, I think, best annotated genes in that interval. So just to conclude, all right, there's many things we could say. How are we going for time, Mr. Chairman? Is it okay? All right. So just to conclude, I think functional genomics can do a lot for us. Genomics and phenotyping together really does provide us with exciting opportunities to really start to make accelerated improvements in crop yields and abiotic stress tolerance in a range of systems. And these salinity happens to be the one that I primarily focus on. We're discovering mechanisms of salinity tolerance in a range of crops. I actually showed you durum wheat and rice, which aren't even on this list. We're also working with these other species. We're using wild relatives to find novel tolerance, and we can access and unlock wild relatives now thanks to the power of functional genomics, the genomics and the phenotyping coming together. We can find genes in the wild relatives that give high salinity tolerance and see if we can integrate them or gene edit these in commercial crops. But also, I think functional genomics gives us opportunities now to domesticate wild species that have very high abiotic stress tolerance. And I know this is a long-term type of program, but I think in universities in particular, it's our responsibility to take these challenges on because we can afford to do that, and I think we should. I'm going to skip over this as well for the interest of time. I'm also commercializing this. I've established a company where we're bringing together plant science and engineering to develop saltwater-based agricultural systems this is a photograph taken this morning as I cycled past of our first pilot stage greenhouse where we're using seawater to cool the rooms, and we're growing the plants in diluted seawater. The roof is cleaned using technology with no water. So we end up with a system where as much as 95%, certainly 90% of the fresh water is substituted with seawater. So this is just trying to develop sustainable solutions for growing plants and commercializing for real, not just talking about it. We are moving into open crops, in particular with grafted tomatoes, using some of our wild relative tomato research. So I'd just like to thank my collaborators, my funders. We've got some wonderful people we work with. Thank the people who have been in the lab in the recent past and the current people in my laboratory. We've got a great group of people. Here they are last year when we last gathered together at our end of year party on the beach here at Kaus. I'd like to thank you all. This is the way we meet now. Yes, isn't that sad? I'd like to thank you all in the audience for your attention. Thanks a lot.
Okay, thank you, Mark. Thank you for a very great presentation.
My pleasure.
And we will have the questions at the end of all the 5 speakers. And I request the audience to kindly send their questions in the chat box. And then we move on forward while thanking Professor Mark Tester again. We move on to the next speaker. And the next speaker for today's webinar is Dr. Hema Bindu. And I have pleasure in introducing Dr. Hema Bindu. She's a senior scientist at ICRISAT and has an expertise in the area of genomics and molecular breeding. Has been working with Professor Rajiv Washner's group for quite some time. And she is working particularly on chickpea and pigeon pea, 2 very important legumes, particularly with regard to increasing production in India under abiotic stress conditions. And Dr. Hema Bindu is a Young Scientist Awardee, medal winner of the Indian National Science Academy, which has a lot of value for young scientists. So I request Dr. Hema Bindu to make her presentation, please. Dr. Hema Bindu, screen is yours.
Thank you, Professor Bansal. I hope you all can hear me and see my screen. Thank you. Thank you for the nice introduction, Professor Bansal. And I'm very thankful to National Institute of Abiotic Stress Management and Alliance of Interbiodiversity and CIAD for this for inviting me to this wonderful webinar. And I'm happy to be part of this team. And also I would like to thank Dr. Mark for setting up the stage and mentioning that functional genomics can do a lot. So I begin my presentation here with the topic integrated omics approach, which reveals drought tolerance mechanism in the case of chickpea. So to give a brief outline of what I'm presenting today, so I would be briefly mentioning about projected impact of climate change, and then brief background with giving an example in chickpea, what is the current status, why do we require integrative omics approaches, and strategy to unravel the drought tolerance mechanism. Again, giving example from chickpea, and findings from transcriptome, proteome, and metabolome approaches. What is the outcome of this entire study or the project? So coming to projected impact of climate change. So globally, at global level, we have several increase in temperature throughout the globe, and whereas in India it is huge. As you can see, all these red indications are so it is very highly vulnerable, all these red marks, and then as you go down, the orange one and the green ones are moderate and low, and the black one indicates very low. As you can see here in this map, it is very, very high in the case of India and also throughout the world. So agriculture needs to produce 50% more food to nutritionally feed 9 billion by 2050. So as you can see here, the world population is raising to 9.7 billion by 2050. What we'll be eating if it is the same in the course of 2050? So instead of food, we need to eat the microcapsules or the medicine. So instead of going to that, we should always produce more food for the increase in population. In this respect, I would like to touch upon the importance of cereals, roots, and pulses. So here comes the importance of cereals, roots, and pulses. There are more than 1,500. They play a very important role, whereas sugars play very less roles, and dietary calories per day, and then vegetable oils, meat, dairy, and others. So as you can see here, it clearly indicates, the red circle here indicates the major daily calories per person by type of food, which is the cereals, pulses, and roots are the major components here. So why do we need this multidisciplinary approaches for crop improvement in this changing climate scenario? For example, if we only go through the phenomics or any other omics approaches or one approach, we are not very clear about the mechanism, which mechanism is playing an important role in order to combat this abiotic or biotic stresses. So hence here we need multidisciplinary approaches, and then go ahead with all these integrated approaches. And then, as you can see here, integrated omics approaches. So genomics and under this integrative omics approaches, metabolomics, proteomics, and transcriptomics play a very important role. And why did we use this? Is we have identified some genes, for example, key genes which are responsible for drought tolerance mechanism, and then some key pathways which are responsible under drought tolerance and drought susceptible conditions. We exactly do not know whether all these genes encode a protein which is also playing a major role in the tolerance mechanism under drought conditions. Hence, we have used this integrated omics approach, combining the transcriptomics data, metabolomics, and then proteomics data together with the phenomics approaches. So we have screened this large population using LYSI-scan at ICRISAT. LYSI-scan facilitate ICRISAT not only in the field but also the phenotyping protocol used at ICRISAT. And then through systems biology, we have integrated all these mechanisms, and we have predicted the phenotype. And then we have identified the genes or candidates which could be used for breeding in order to breed the superior lines. So chickpea is the third most important protein-rich food legume crop, which is covered over 40 geographically diverse regions, and it is a diploid and self-pollinated crop with 2n is equal to 16. The genome size is approximately 740 mbp, and almost 74% of the genome is made available in 2013 by Dr. Vashni's group at ICRISAT, and the production is on an average it is 14.8 metric tons. And as you might have already known, the highest producer and importer is India. Also, I would like to make a brief mention that there are two types of seeds, Desi and Kabuli. The Desi seeds accounts for 80%, whereas Kabuli accounts for 20% of the chickpea. So the major chickpea producing countries are mentioned here: Australia 6%, others there are 5%, America 4%, Africa 4%, Turkey and Pakistan. And India, as you can see here, it is 67% of the total production throughout the world. So to give a brief background on omics approaches to study stress responses in crops, we have published this paper in 2013. And then once we develop different genomic resources coming from different genetic resources, and then through two different stresses, like whether it might be drought, salinity, or any other insect or pathogen attack or something, and then you sequence the genome or transcript, generate a transcriptome assembly, and then you study the gene expression analysis through different analytical methods or tools, and you identify the candidate genes. Once the candidate genes are identified, either you clone the gene and predict the function of that particular gene using several approaches like gene silencing, overexpression, or tilling, or you can go towards the marker development and identification of linked markers to that particular trait of interest. So, and then you go for marker assisted selection and evaluate the lines in the field once you develop the lines. So ultimately our aim is to develop improved or new cultivar in this study, or through our process or pipeline. So in this pipeline, we have generated a chickpea gene expression atlas, where we have generated the transcriptome reads from different or important stages of chickpea, which covers the entire life cycle of the plant. For example, here germination, seedling, vegetative, reproductive, and senescence. We have generated the sequences from 27 tissues of different stages, and studied the expression profiles at each of the stage, and also comparing the expression patterns of each genes at different stages throughout the life cycle of the plant. So this has been published in 2018 in Plant Cell Environment. And we to mention here, I would like your kind attention. We have used the genotype ICC 4958, which is a drought tolerant variety here, to generate the chickpea gene expression atlas. So we have identified through this study several important genes which are responsible for growth and development, seed development, nodulation process, and root development. These candidate genes are being taken forward for further studies. I would mention in brief about these in my coming slides. Next, what we have done or how we have planned this study, and what approach we have followed. Initially, we have in this study we have taken 4 genotypes. As I have already mentioned, ICC 4958, which is a drought tolerant parent, and what we have also taken as a sensitive parent, ICC 1882, which is a recipient parent and also high yielding. Here, in order to study highly tolerant and sensitive parents is a routine study. What we have done is we have included another elite cultivar, which is also tolerant for drought, JG11, and we have crossed this tolerant parent with another tolerant parent, JG11, and generated near isogenic lines, JG11+. This has been done at our breeding unit at ICRISAT, which is led by Dr. Puran Gaur, and Srinivasan right now is taking care of the breeding unit of chickpea. So we have used these lines together with these elite parents, and planned this study, and I have conducted this study for integrative omics approaches in the case of chickpea. So what we have done is we have taken up these lines we have selected these lines using molecular or genomic approaches. And then we have done transcriptome analysis in all these 4 parental lines together with near isogenic lines. We have done sequencing with both well-watered and also drought-tolerant conditions. So we have used two conditions. One is well-watered and another is water stress. So in case of all these 4 lines, we have used this and then generated the transcriptome data. Also, the same tissues were used for generation of proteomics data and metabolomics data as well. We have integrated the genes or the analysis coming out from transcriptome and then proteome and then metabolome. And we have studied candidate genes which are common for all the 3 and which were playing an important role in the drought tolerance mechanism. So the candidate genes were identified, some molecular markers which were very well linked to these traits were identified, and the metabolic pathways unraveling the drought tolerance mechanism were also identified. I would be talking more about this in my coming slides. And these particular markers or the candidate genes are now being used in the breeding program for marker-assisted selection, or we are going ahead with genetic engineering. So ultimately, we have combined this data with several physiological data or phenomics data, and we have phenotyped all these lines under our phenotyping facility available at ICRISAT. And then we have also identified key candidate genes which were linked, which were present in the QTL hotspot region, which were responsible for more than 13 or 14 different traits for drought tolerance mechanism and different yield and yield related traits. So this QTL hotspot plays a major role in controlling the drought tolerance mechanism. So what we have done is we have taken up data analyzed from all the 3 approaches, the transcriptome, proteome, and metabolome, and identified what genes are exactly lying within the QTL hotspot region. So we have applied these molecular breeding strategies for improving the drought tolerance mechanism, drought tolerance in the case of chickpea. So as I was talking to you, we have taken up different 5 replications of each of these lines or genotypes under well-watered and drought-tolerant condition at the transpiration ratio 0.01. So drought was imposed and drought stress was imposed under glasshouse conditions, and then we have at transpiration ratio of 0.1, the root tissues were collected from each plant or at different replications and used for all these sequencing. So for transcriptome, we have used RNA-seq using next generation sequencing technology, and for proteome, we have used liquid chromatography and mass spectrometry, and for metabolome, gas chromatography and mass spectrometry has been used. These technologies were used for these 3 approaches. So using these, as I have mentioned, we have ICC 4958 control and stress, and similarly JG11 control and stress here, JG11 plus, which is near isogenic line, control and stress conditions, and 1882, which is a sensitive parent for drought. So we have used control and stress. Control is nothing but well-watered, and stress is for drought stress. So approximately 364 million raw reads were generated, and then we have filtered 341 filtered reads using from this raw reads, and these reads were mapped onto the chickpea genome. Around 304 million reads were mapped onto the chickpea genome. So approximately 89 to 90% of the reads were aligned to the chickpea genome. So when we analyze the transcriptome, global transcriptome and proteome data, as you can see here, all the JG11 samples were clustered together, and ICC 4958 and 1882 were clustered together. Unlike in the case of proteomics data, all the control samples of different lines were clustered together, and all the stress samples were clustered together. So we have identified the phenotype and treatment specific identity, And in the case of proteomes, it revealed a treatment-specific biological identity, that is control and stress, as I have already mentioned. These tissues clustered together were control or stress specific irrespective of the genotype here. However, in the case of transcriptome, they are genotype specific rather than the treatment specific. So when we have conducted the differentially expressed gene analysis, we have identified several genes which were transcriptionally active under each condition of for each genotype. And then we have also identified the genes which are which showed low or moderate expression, and genes with high expression were all identified. So we have identified common genes and unique genes as well. For example, as I have mentioned here, around 3,956 unique set of differentially expressed genes were identified in all the 4 genotypes studied, and 2,000 genes, around 2,000 genes were common for 2 or more genotypes. So similarly, we have also identified unique genes which were specific to one genotype, that is ICC 4958. For example, here we have identified 717 genes for ICC 4958, 862 for JG11, and 358 for JG11 plus, which is an ER isogenic line, and 400 genes approximately for ICC 1882, which is a susceptible parent for drought. So we have also identified 289 differentially expressed genes which were common for all the 3 genotypes. So our idea here is to identify common genes and also genes specific only to ICC 4958 and JG11 plus. Why we are looking for these 2 particular genotypes is the QTL from 4958, QTL hotspot, which I was talking to you earlier during my presentation, has been transferred to JG11, resulting into JG11 plus. So we are we assume that this near isogenic lines is more tolerant either than ICC 4958 and also JG11 plus. Hence, we try to see which genes are common for ICC 4958 and JG11 plus, because those particular genes which are specific for these 2 lines could be from the QTL hotspot region. For example, here you have a pictorial representation of these candidate genes which were identified on each of these chromosomes. The QTL hotspot lies on linkage group 4, and we have identified several key candidate genes responsible for drought tolerance mechanism on this particular chromosome. So the prediction of inheritance of drought tolerance in JG11 plus based on transcriptome as well as proteome analysis. And we are trying to look for the inherited differentially expressed proteins from ICC 4958 and JG11 into JG11 plus, which is an ER isogenic line mapped onto different Chickpea chromosomes. So for expression analysis of these particular DEPs for select combinations, for example, as I was talking to you, we try to identify genes which were expressed only in ICC 4958 and JG11 plus. So these key candidate genes which were identified in these 2 genotypes were highlighted here, and which were common for all three tolerant lines, that is JG11, ICC 4958 and JG11 plus. And here we see the genes which are common for JG11 and JG11 plus, the parent of one of the parent of this particular near isogenic line. So we have identified 15 differentially expressed proteins, 9 DEPs and 36 DAPs respectively for these particular combinations. And also we have studied what are the correlations between the DEGs identified from the transcriptome and the versus the DEPs identified from the proteome. For example, here you can see the JG11 and JG11 plus, you have 2 genes which were correlating between the transcriptome data and the proteome data. Similarly, in the case of 4958 and JG11 plus, and in the case of DEGs and DEPs, you have 6 genes which were identified. For all the 3 tolerant lines, we have identified 2 genes which were correlating from transcriptome and proteome data. So integration of proteome and transcriptome data has been done to identify variations across drought responsive proteins and genes. A one-to-one correlation analysis is identified, and 900 proteins out of 2751 proteins which were identified from the proteome data had their corresponding genes in the transcriptome data. So as I have already mentioned, 2 out of 36 DEPs and 6 out of 15 DEPs coming from 4958 and JG11+, and 2 out of 9 DEPs coming from all the 3 genotypes were identified as correlated genes between the transcriptome and the proteome data. So these are few pathways which were identified, which were significantly enriched. There were 75 in total, representing 175 enzymes, and these were involved in more 111 pathways. These specific pathways were identified, and key candidate genes which were differentially expressed both in transcriptome and proteome, together with correlated data from transcriptomics and proteomics, were identified in these metabolic pathways, and these metabolic pathways were highlighted. And then coming to the metabolomics data under drought, stress, and control conditions, there were a total of 34 metabolites were identified and were highly reproducible among these 3 biological replications in 4 different cultivars which were studied. So the control and stress conditions were clearly separated, which explain 34.97% of total variations. So this indicates how we can demarcate the control and stress conditions under this study. So drought imposition has considerably increased the accumulation of amino acids such as proline, valine, threonine, serine, aspirin in all the cultivars studied, in all the 4 cultivars studied. So when we have done the clustering, as you can see here, the control and stress samples of all the tolerant lines were clustered together, irrespective of treatment. However, JG11 plus control sample has overlaid separately, and then here ICC 1882, the stress and control conditions clustered together. So variation in all these metabolite levels in root samples of chickpea genotypes under control and stress conditions is well illustrated by clustering heat map analysis. So as I have mentioned, 3 key metabolic pathways were identified under this study: the galactose metabolism, abiotic biosynthesis, antibiotic biosynthesis, and isoflavonoid biosynthesis. So the 10 proteins which showed the correlated expression both in the transcriptome data as well as the proteome data, and also which showed the 6 metabolites which has enhanced correlation with these pathways were identified: the galactose metabolism, antibiotic biosynthesis, and isoflavonoid biosynthesis. So the key genes were identified among these 3 pathways, and these particular key genes or proteins which were identified are being taken forward for further studies and confirmation. So the take-home message from this study is, so we have explored the transcriptome, proteome, and metabolomic data analysis of 8 tissues from contrasting drought-responsive genotypes, capturing all important aspects of drought tolerance mechanism in the case of chickpea. So comparing the gene and protein expression patterns at different treatments and genotype specific genes and proteins were identified. Furthermore, expression patterns of correlated genes and proteins were identified, and thus illustrating in the identification of potential candidate genes for drought tolerance mechanism in the case of chickpea. So, and also metabolite profiling identified changes in the metabolites under drought tolerance conditions. So these key pathways have also been identified, as I have mentioned in my previous slides, which are involved in drought tolerance mechanism. So finally, what I conclude is there's all the data analysis coming out from this transcriptome, proteome, and metabolome we have integrated these data sets, and also this integration of omics approaches provided molecular insights into the drought tolerance mechanism in the case of chickpea. So it provides, this multi-omics approach provides a strategic research framework for faster data-driven advances in crop improvement for current and projected climates. Finally, I would like to acknowledge Dr. Rajiv Vashni, our group director, Genetic Gains Program, Anu Chitneni, our manager, Rutvik and Amir Khan, PhD students with Dr. Vashni, and Sandeep, who have helped us in analysis. And also Dr. Wolfram and Arindam Pallak from University of Vienna. I would also like to acknowledge the funding source from Department of Science and Technology, Government of India, and also research program on genetic gains and grain legumes and dryland cereals. Thank you so much for the opportunity, and I would be happy to take up any questions.
Okay. Thank you, Dr. Hema Bindu.
Thank you, sir.
For a nice and an elaborate presentation. And we move on to the next presenter. And until a few minutes ago, we were wondering if Dr. Matthew Reynolds has already joined. Yes, I'm very happy to see him there. And the next presenter is Dr. Matthew Reynolds. And a pleasure in introducing you all, Professor Dr. Matthew Reynolds, who is again a very well-known, world-renowned scientist, actually speaking a plant physiologist in the area of drought and heat tolerance, particularly in wheat, and working as head of the physiology section or department at CIMMYT, in Mexico. His research actually has been quite innovative, I would say this, with regard to drought and heat tolerance in wheat, as his research has helped in creating new generation of advanced wheat lines, based on physiological breeding approach, which is a kind of a novel approach which he introduced, globally speaking, for selecting, some of the genotypes which are tolerant to drought or heat tolerance, particularly in wheat, and are being applied, I know, in other crops as well for adaptation, particularly to drought and heat stresses. These advanced lines are at the verge of also at times, being released as varieties by breeders. As I was talking to you, there was not a single trait which we could depend on for selection, for the breeders, but then Dr. Matthew Reynolds' research has given a lot of leads, as a physiologist into this area of research. And he has developed all these methodologies, particularly for high throughput phenotyping of a large population, large germplasm collections actually at a single site. And he has also, taken out several, I would say, manuals, and is helping globally many of scientists, including me. I was one of the beneficiaries while visiting, his lab at one time in Mexico. And he is, more importantly, at the global level, is coordinating a heat and drought, wheat improvement consortium, HEDWIG, actually we call it. I never pronounce the full name. HEDWIG is quite famous and well known, and that is the Heat and Drought Wheat Improvement Consortium, and which is contributing again quite significantly to train the young scholars and postdocs and people around in the world in his lab and also at their respective places. So friends, it's a pleasure to welcome, and I'm very thankful to Dr. Matthew Reynolds for accepting our invitation. As a good friend he's been collaborating with us for quite some time together. And I now request Dr. Matthew Reynolds to make his presentation, please. So Dr. Matthew, the floor is yours. On the screen. Thank you.
Thank you very much, Dr. Bansal. That's a very nice introduction. Let me share my screen.
Please.
And we can make a start. Well, so what I want to present today is an overview of the new project that will underpin HEDWIG. And this is a new source of funding for the CGR, in fact, from the FFAR. You can see the logo at the top left. And so putting this together, Shiva and others were involved in designing basically a series of activities, a strategy to help us improve the climate resilience of wheat. And you can see what we have here is a table showing the goals and how they relate to the research gaps and the main outcomes and beneficiaries. But just to keep it simple, I'll just run through the goals, the gaps at the top. that we'll address, and I'll come to, and I'll mention briefly. Oh, by the way, Dr. Bansal, how long do I have? What's my timeline here? I should have checked.
30 minutes.
30, okay.
Sure.
Okay, I'll make
Thank
a
you.
note of that. Thank you. So, so among the research gaps are insufficient detail of breeding targets. So basically details about the environments, the stress profiles, and how genotypes will interact with those. The limited genetic diversity for climate resilience. As a matter of fact, that's how Dr. Bansal and I first met in the context of screening Indian wheat lines for heat tolerance when he was director of the germplasm bank. So getting some more of that diversity into modern elite lines. The need for more comprehensive phenomics tools. So that's a very fast-moving field. So a lot to do there. The limited understanding of genetic bases of climate resilience. That is something that we all recognize, since so many of the traits associated with heat and drought tolerance are complex traits that are, although there are many studies showing regions, MTAs and so on, our actual ability to move those genes around is limited in breeding. The need for smarter strategies to stack traits. So when you're putting complex traits there, we call this physiological breeding. If you move these, when you're moving these traits around, they can be a bit slippery. So we need better strategies. So that's one of the activities Shiva will be leading. And then some of the other activities that are mentioned here, these relate more to the link to the breeding. So improving the genetic gains, and of course, the use of the International Weed Improvement Network to scale out technologies, whether those technologies be new germplasm or phenotyping tools. As Dr. Bansal mentioned, these are all useful outputs. Then there's the issue of the bottleneck between basic plant science and application in breeding. This is a huge issue. It's where we need more translational research to get proofs of concept. There are many publications that claim that findings could have huge implications for plant breeding, and yet that final proof of concept is typically omitted because there's a kind of a funding gap. So a lot of our emphasis is on showing these proofs of concept through pre-breeding and international testing. And of course, insufficient capacity in many national research systems through training and other kinds of activities. So that, some of you may have seen this before because we published it a few years ago. It's what I just showed is not that different to this idea of a sequence for pre-breeding, where you start off with a crop design, you find genetic resources and phenotype those to pick the best ones. You make the genetic analysis in order to be working with panels, in order to identify MTAs, and also antagonism even potentially between regions, trait compensations, and understand better their genetic base. You have then a lot of information to make crosses, but it's still a big part of it is a numbers game, notwithstanding the ability to select parents for complementary traits and the high throughput phenotyping of progeny. And then of course our international network, which puts that improved material in the hands of breeders, which can be used for crossing. It can, in occasional instances, lines have been released from pre-breeding, and also, of course, some useful material for research. So the first goal that I mentioned was the lack of or insufficient understanding of our target environments, and here we're taking advantage of the data set from the International Wheat Improvement Network that's been going for 4 decades, coordinated by CIMMYT. And so what you have is concepts of germplasm grown over many environments. In fact, the network distributes about 1,000 new lines every year. And a little under half of that data is returned and can be used for these kinds of analyses. And we have a group in Henan who are leading this effort. I have a postdoc, Margaret, with us, who's also looking. She has a particular interest in yield stability. And there have been studies using this, this data set. The breeders have shown a number of retrospective analyses of genetic gains. And there was one particular study led by modelers, led at Florida and other places, that were showing the likely impact of warmer temperatures in a number of regions. And we all know that these are quite serious concern. But there's a lot more that can be done with this data. And so this is one of the, let's say, as I mentioned, the first gap to fill is to try and get a better understanding of this. So we'll be using historical data, but also sharing a trial. I'm hoping that some of you will involve in that, and looking for yield response and G/E under different heat and stress profiles. And of course, part of the design then for when one has defined better the environment, it makes it more easy to use the traits. We know that there's always G/E and even background effects when it comes to putting these traits together. But certainly by having a better definition of the target environments, it makes it more easy to determine which traits are more likely to have a benefit when you design the strategic crosses. We can come back to this if there's time at the end. So in terms of the second gap is finding more and better sources of heat and drought tolerance. And at CIMMYT, we're lucky to have right there the World Wheat Collection that includes over 150,000 genotypes from more than 100 countries. That's the biggest unified collection available anywhere. And we, over the years, have spent time screening that in the field. 70,000 have been screened in this way, but these are huge numbers. Dr. Bansal and I discussed that when I visited. It's hard to be extensive and intensive. So we start off with a big number, and we rely largely on very simple traits like emergence and the ability to stay green, visual estimates of biomass. We did actually measure yield on 70,000 under heat and drought. And in order to essentially, here's an aerial photo showing the small plots that we sowed. Eventually you come down to diverse panels of diverse but promising looking material that we can apply in research and breeding. And 70,000 was a lot of work, back-breaking work, as you can see in the photo. And still only a small fraction of what's available in the collections around the world. It's currently estimated at around 0.8 million. So a lot of opportunity there to explore new sources of climate resilience. Now, as I mentioned, outputs from this kind of work can give you a number of panels. So here we see a bred wheat diversity panel and a synthetic panel. And when you, sorry, I'm having a problem here with my, let's see what's going on. Too many windows. I'm frozen. Let's see, what's this about? Sorry, everyone. There's I'm not sure why my slides are not advancing. Here we go. Okay. Here's the synthetic panel. We picked 160 based on screening of 2,000 that were made. So these are brand new genomes, resynthesizing the hexaploid wheat. And that's quite an interesting set of germplasm. If you look at this graph, what we're seeing is a plot of the grain yield against the biomass of these primary synthetic lines under heat stress. And the colored dots are elite lines, and you can see quite clearly on the y-axis that there's not really any primary synthetics significantly better than the best checks. But what you see on the x-axis is about a half of the synthetic lines are doing much better in terms of total above ground biomass than the elite checks. And that biomass, obviously the primary synthetics are not adaptive. That biomass potentially could be partitioned into yield. The fact that you have high biomass tells us that these lines have the traits that are adapted to heat. The radiation use efficiency, which could be associated with many different biochemical processes, but they're obviously coping with the heat. It's just that they're not adapted to converting it into yield. So that material right there with this kind of data, obviously we picked some of these for crosses, but more study will tell us what traits they have and how can they be used in a more complementary fashion. There's a lot of potential right there just in the primary synthetic material. And you see a similar kind of phenomenon when you look under heat drought stress, as well as even at high yield potential, you also see that higher radiation use efficiency. Shiva made this this map that shows the markers associated with some fairly common agronomic traits. And what's interesting is that the markers are typically not coinciding between the bred wheat panel and the synthetic panel. Now, on the one hand, you could say that that's a bit of a problem. It shows us the complexity of some of these climate resilient traits. But on the other hand, it's an opportunity because what we're seeing in different regions that can potentially be combined. So a lot more needs to be done on that, obviously. Just finally on using genetic resources, by fingerprinting the genetic resources, and there was a big effort under the Seeds of Discovery project, one is able to distinguish distinct groups quite clearly. You can see here in the green the lines derived from synthetic hexaploids. You can see the primaries in pink. You see land races in blue and light blue elite cultivars. And this is a tool that is being used, and we have used in this new project with the FFAR, to make sure that when you start building new panels, you can pick, rather than be all in one part of that figure, you distribute your selections across it so that your panel represents a good range of diversity. So that's a nice use of molecular fingerprinting to help us with a more comprehensive screening, or rather a more, what would you say, a more strategic screening of genetic resources. And I recommend taking a look at the Sansolini publication that just came out recently. So, third gap, improve phenotyping methods. And we're looking here at a drone and a vehicle, where it says number 4, there's the drone. Number 5 is a vehicle that one can push through the plots with various sensors attached. Obviously, the payload of a ground-based vehicle is a lot higher than a drone. And these are alternative, just a couple of alternative approaches for high throughput phenotyping. there are many more restrictions these days associated with flying drones. So it's good to be able to count on different approaches. Drone is fantastic, obviously, when you've just irrigated, but the handheld can typically carry, the cart, ground-based cart can typically hold more sensors. So still much more high throughput than going through with individual instruments. And when you're talking about measuring thousands of genetic resources from collections, you absolutely need these high throughput approaches, and they need to be carefully calibrated, and you need good, if you're using images, good image extraction approaches. But with these, we're able to get fairly comprehensive data on some fairly wild and woolly genetic resources. These same tools also obviously apply for progeny screening. So between thermal images and spectral indices, there is quite a lot of information you can get from a plant without making a single destructive measurement. However, we don't discount destructive phenotyping, the classical growth analysis. There are certain traits that we have found to be associated with yield, particularly yield potential, in fact, traits that might express around flowering that are associated with partitioning, fruiting efficiency, and so on. And then, of course, the roots, these all need to be measured with more precision or more destructive approaches, as you can see there in the photo. And so we must consider both of these. I consider that in general, the precision approaches are very good for when you have boiled down a larger collection of genetic resources into what you might consider a candidate crossing block. The precision approach is very good for picking parents that can be crossed, because you're dealing in the hundreds. But if you are screening large collections of genetic resources or thousands of progeny, that is when the high throughput approach is absolutely necessary. Otherwise, it's just really not feasible. And I'm sure everyone is aware that we particularly favor canopy temperature under drought and heat, in fact. It is a tool that non-destructively and very simply can predict yield under these two stresses with a pretty high level of precision. The graph is from a PhD student, Juan Jose Olivares, and it shows how robust the trait is, that it will show a strong association with yield under drought, explaining over 60% of variation. This is in the real population, whether you're measuring before or after heading, or in the morning or in the afternoon. So it is a very robust trait. And further research, this was led by Marta Lopez, showed that that trait not only predicts yield, but actually predicts very difficult to measure trait, which is root dry weight below 60 centimeters. So on the x-axis, we see root dry weight between 60 and 120 centimeters. And on the right-hand y-axis, we're seeing canopy temperature, and you're seeing an almost perfect correlation between root dry weight at 60 to 120 and cooler canopies. That's an R squared of 0.88. That's very high. And that is very encouraging, because being able to estimate roots at depth with a very simple to use apparatus like an infrared thermometers represents kind of a breakthrough. Even though this is not particularly recent research, we are following it up in root phenotyping in the field. The alternative is to go into the field with this kind of hardware and dig out soil cores, which is so much more labor intensive. At least 100 times more effort is required to understand root depth using that method compared to the simple measurement of canopy temperature. We believe there's a lot more promise to get out of that, and we're actually working currently on developing a dedicated index for root characteristics using remote sensing. Now the genetic dissection, I'll just give one example. This graph shows us how night temperature is determining yield, farmer's yield. This is in the Yaqui Valley, where we do our research in Oregon, in Mexico. And there's a very, very tight relationship between the night temperature or minimum temperature and yield. And that correlation is actually much stronger than it is with the maximum temperature. And that's telling us that we really don't know enough about what's happening at night and why the plant is responding so much more to cool, to cool temperature. And so we had a data set from the WAMI where we had temperatures during the day and the night. And so Shiva got to work analyzing that and came up with this very nice analysis that indicated indeed that not only, as in the previous graph, you see a main effect of, you are seeing one genotype on the red line that did better, but still affected negatively by night temperature. This analysis is showing there's a lot of G by E for this, and it especially in relation to the minimum temperature. So the grain yield was actually very significantly affected by the G by E grain yield was very significantly affected by minimum temperature at 3 growth stages, but particularly around heading and the grain filling stage. Whereas the maximum temperature, you see almost nothing. So that's very promising, and Shiva went on to make a GWAS and identified one gene that was known associated with grain weight and 2 novel regions that based on the slopes of each genotype as they respond to minimum temperature, which is a very good basis for follow-up, and that's work is currently being followed up. Okay, so coming to the pre-breeding, this pyramid indicates more or less our overall concept for how to make strategic crosses. And if you think of yield at the top of the pyramid and the genes as the foundation, what you have between is many of the traits that go into generating yield. So on the left, we have biomass, on the right, we have harvest index, but each of those are comprised of different components. So for example, biomass is affected by radiation use efficiency, which has a number of other components like spike photosynthesis, the canopy architecture. If you get down to the cell and subcellular level, the rubisco, respiratory metabolism. And then you have the light interception, where you have tiller dynamics coming into effect, early vigor, the retention of chlorophyll. If you look on the partitioning side that gives us the harvest index, there are characteristics associated with both grain set and grain size. And so we developed this pyramid as a kind of conceptual model to help us design crosses so that we combine as many of these components of yield as possible in a single cross, with the idea of achieving cumulative gene action for these traits. And in fact, for yield potential, we're working on an even more sophisticated version of this, which we hope to publish soon, where we've developed a wiring diagram where we show connections between these and many other traits, in fact. But anyway, that's coming up later. So just some results from the pre-breeding work. This is the results of the satin grown in 33 environments in last season. And what we see is that the best 3 lines from the strategic crossing approach, they have about 7% more yield on average than over the local checks. And if you take the very best line from the strategic crossing, we see a 4% advantage over Simit's best check batch. So while these lines may not be ideal in terms of their disease resistance profile, although most of them have rust resistance. And they may not be ideal in terms of quality. They certainly are showing yield potential. So these are the kind of lines that can be picked up by your breeders and crossed with, or put into variety trials if the other characteristics are suitable. So, as I mentioned, although pre-breeding is not set up really to release varieties, there have been some successes over the years from our pre-breeding approach. And it's interesting to see, when you look at the pedigrees, how the genetic diversity has been brought into many of these examples. So, for example, MEX 9427, that's a Mexican landrace. Sokol is a synthetic derived line, which is in all of these, in fact. And again, we see two Mexican landraces in this line, Quinoa, that was just released in Afghanistan. One from the state of Mexico and one from the state of Puebla. And there in the middle, Sokol, a very successful synthetic derived line. So it is possible, the proof of concept is there, that pre-breeding with some fairly exotic genetic resources not only can get you right up there with the best elite material, and in some cases give you a yield advantage, but without any of the linkage drag that is typically associated with these kinds of materials, and which often has traditionally put breeders off from using them in their crossing programs. So certainly an encouraging result. Okay. I don't know why I'm having these problems to deal with my computer. I apologize. There we go. So I won't spend much time because my time's coming to the end. Part of our program, as I said, our new program was required to link it with the breeding, so which is a great thing, but is not specifically the area of physiology. But there are a number of continuous improvement in breeding objectives that have been agreed on with the Gates Foundation and our breeders, Ravi Singh and his team. And I just want to mention one of the results recently from Philomene Juliana, where she made an exercise of looking for key markers associated with important agronomic traits, used that to phenotype almost 50,000 lines from the global breeding germplasm pool, and demonstrated that this change in frequency of key alleles in different chromosomes here. And that's a fascinating result, I find, recently published in Nature Genetics, that is showing that many of these markers are on their way up in terms of frequency in elite material, or some have saturated and can be considered fixed. And a great way to monitor progress and indicates kind of genetic improvement in a tangible way in the CIMMYT elite material that's being distributed every year. So in terms of dissemination, we rely very heavily on the IWIN network. So as I say, it's not just the technology can be frustrating. It's not just the germplasm, but other technologies like phenotyping and so on. We can use the network of IWIN, or the more restricted Hedwig network that Dr. Bansal referred to earlier. And it's very much a network that is multi-directional. So germplasm is shared, phenotyping ideas are shared. And we try to work on common platforms where we can in Obregon. But there's also, for example, under IWIP, winter wheat platforms being established, hopefully more heat and drought platforms can also be established, places where people can come together, work on common germplasm, and improve the comprehensiveness of our understanding of adaptation to heat and drought. Sorry, I should have mentioned here that we shall be looking for ideas actively. We have a small fund available, and I will be sharing a call for that so that people can actually come together at the platform in Obregon. And there are many other outputs that one can expect from this kind of network, apart from germplasm, the knowledge, one of the key goals is the training of the new generation of scientists. They're the ones who are going to have to really face the harshest effects of climate change that we already see, but are predicted to get a lot worse. Here's an example of the manuals that can also be shared through this kind of network. So our take-home points, just to finish up now, climate is certainly becoming warmer and less predictable, but many opportunities do exist to improve wheat's adaptation. The advances in phenomics and genomics, the opportunity to explore the vast untapped reserves of genetic resources, and new approaches combining physiological and molecular breeding to put together the traits and alleles in a very targeted way, and not just targeted for heat and drought generically, but the more we learn about our selection, our target environments, put together in a more regional or local way to conform to certain types of heat or drought profiles, soils, and so on. And of course, that the fact that impacts will reach farmers and consumers much sooner if we try to coordinate our efforts through networks like Hedwig and the Weed Initiative's Ahead program. So thank you very much, and if there's time, I'm more than happy to take some questions.
Okay, thank you, Matthew Reynolds, for an excellent presentation, covering various topics as a physiologist, right from phenotyping to genomics to molecular breeding. It's certainly a very good, useful resource for all of us, and I'm sure for all the participants. So thank you, Matthew, again. And so we move on to the next presenter. And we have two more presentations, actually, both from our national system from India. And just for the matter of information to the participants, you know, that we have a very strong national program, actually speaking, in progress in India, particularly to focus on abiotic tolerance in various croplands. We have Professor Sudhir Sapuri's lab actually working in JNU and now in ICGEB, one of the international centers on genetic engineering and biotechnology. We have Professor Akhilesh Tyagi, and one of the old players we have actually Dr. Anil Grover, you know, from Delhi University South Campus, who has been contributing quite significantly in rice. In rice, actually, we have quite a lot of effort. And apart from the institution, you know, organizing this, Dr. Himanshu Pathak's institute, we have few other institutions of the ICAR and several national international labs working in India, you know, to focus on abiotic tolerance, because India needs all these technologies more than probably any other country, because we have large tracts which remain unirrigated or rain-fed. We don't have those facilities of irrigation. We have a large tracts which are affected by salinity, and now with climate change also high temperature. And so therefore, I'm sure we are going to benefit a lot from all these presentations. And moving on to the two national speakers, the first of them is from India, Dr. Girder Pandey. And I have a pleasure, and thanks to him again for accepting our invitation. And a pleasure inviting, you know, and introducing you, Girder Pandey, who is a professor at the Plant Molecular Biology Department of Delhi University South Campus, you know, and also he's well known for his work on calcium-mediated signaling or functional genomics and stress adaptation plants, and focusing on a model crop like, or model plant like Rhodopsis, and translating that research into rice. He is the winner of several awards. Time will not permit me to go into the details, but one of them, of course, happens to be a National Bioscience Award by the Department of Biotechnology, you know, which is considered to be a good, you know, award, particularly for younger age, relatively younger age participant or scientist working in India. So I have a pleasure in inviting and welcoming Dr. Girder Pandey for his presentation, please. Girder.
Thank you very much, Professor Bansal. Am I audible?
Yes, you are audible.
and good evening to all of the participants and then the eminent speakers. And then, so I'm going to talk about one of the important aspect of abiotic stress, that is the mineral nutrient deficiency, specifically covering some of the aspect of the very important mineral nutrient called potassium. So this is the title, the potassium deficiency impose abiotic stress management in plant, a functional genomic intervention. Basically, it's like more functional characterization. So as I think in the beginning, Dr. Pathak has already introduced this abiotic stresses. Many of the abiotic stresses are creating a lot of problem for the crop productions, and I'm not going to talk more about those. But I just want to give you some brief glimpse of this mineral nutrient. So plant need a lot of these mineral nutrients, for their growth and development. Without these, I think they are not able. The plant is not able to grow properly, and that if they cannot grow, there will be no yield. So there are macro elements, micro elements. I think we all know about this. And one of the very important aspect in the agriculture is specifically because the NPK. When we talk about the fertilizer, covering the nitrogen, phosphorus, and potassium is heavily used in most of the crop plant. So this is, I think, the textbook model suggests that when the plants are undergoing the mineral nutrient deficiency, you can see some visible symptoms. So these are some of the typical visible symptoms seen for the plants when they are undergoing the nutrient deficiency. I am going to talk, as I said, mostly cover potassium, as the nutrient. So let's see the function of the potassium in plants. It's a very, very versatile and important mineral nutrient, because we know that potassium regulate a lot of physiological processes such as membrane potential, ionic and water homeostasis, cell turgor, cell growth. Stomatal conductance is one of the very important areas. Besides this photosynthesis, many of the photosynthetic machinery component or enzymes require potassium for their proper functioning. If there is a low potassium, then you have impaired nitrogen and sugar balance, and there is a problem in the long distance transport. So it is another aspect is the salt tolerance, as Professor Mark Tester said. The salt stress is one of the important problem for the crop productivity. And then I think sodium and potassium has very hand-on-hand relationship with each other. If you in the salt tolerance also, you see a very close connection with the potassium. You see there is the ratio of the potassium to sodium is affected. And as I mentioned, that potassium is a very agronomically important component of this NPK fertilizer. So let's see the deficiency of the potassium. The potassium deficiency usually seen in the older leaves, and it's the symptoms are quite marked. You can see the bleaching or chlorosis of the leaves, and then subsequently there are many problems with the plant growth and development. So yeah, this slides represents, I think, how the potassium. I think this is another representation how potassium deficiency symptoms you can see in the crop plants like tomato, alfalfa, and rice and wheat. So all of them show that there is a chlorosis of the leaves. These are the kind of the early symptoms, and then later on the whole development of the plant is hampered. So if you look at the potassium status in Indian soil, this is, I think, an old slide. It says most of the Indian soil is, also having a lot of problem with the potassium. It's not, most of the reason in the agricultural field are having potassium deficiency. Because potassium deficiency, which represent most of the potassium available in the soil, is in the dissolved form, which is actually available for the uptake by the plant or crop plants. And the remaining, is not available, and you need a different kind of a methodology to make these, this nutrient dissolve into the water system or the rhizosphere, and then the plant can be able to take this. So if you look at the overall, so to mitigate this, as I mentioned that, the crop plant need a lot of application of this fertilizer called NPK. I think this is one of the primary fertilizer, I would say, being added to most of the crop. And the potassium is an important component of this NPK, because I will show you, I have already shown you some symptoms, and I will show you another, some of the phenotype where you see if you grow the plant in the potassium deficient media, then their overall growth and development is basically arrested. So the increase in the, as you see, the increase in the global usage of this NPK based fertilizer is basically over a period of time. It's being recorded, and then now I think we are somehow using this chemical heavily, I mean fertilizer, in the soil for fertilizing the soil for the crop system. So what is the possible way to basically tackle this problem, say for the, and then over a period of time, when the plant or the crops are growing, because this potassium is one of the important component of the dry weight of the whole plant. It is being, the plant use up this mineral nutrient, and then eventually the soil, the dissolved concentration of the potassium in the soil is getting depleted. So over a period of time, when there is a crop productivity or crop is being produced in a repeated manner, then you will see, like any other kind of mineral nutrient deficiency, the depletion of the potassium in the soil happen, and then the soil become potassium deficient or potassium depleted, and then what happen is you need to, it's a compulsory, you need to add the potassium based fertilizer to the soil. Otherwise the plant or there will be no crop productivity is going to be a big problem. So the possible way of handling or this crop, the potassium deficiency, to mitigate this potassium deficiency for the crop system, if we understand how there are many different ways, we can basically tackle this problem. And one of the important ways, if we can understand what is happening at the biological level for the sensing, signaling, and uptake and transport of this potassium. Specifically, overall covering, how this potassium is being sensed and how this potassium ion is being uptaken. And then later on, we also talk about, potassium use efficiency, potassium, and so on. So if you look at the plant systems, typically Arabidopsis is a model plant. There are a lot of potassium transporters and channels. As you can see, these are the potassium transporters and channels classified into different subcategories. And similar number of the potassium channels and transporters are also found in rice. And it has been now, I think, reported, and it has been now known that Arabidopsis and rice has a very similar kind of, the potassium transporters and then the signaling response molecule or component. So now I think, as I mentioned, what is the way, we can understand, what is the way we can tackle this problem of potassium deficiency? So one major way is if we can understand better the whole sensing, signaling, uptake, and transport, which overall comprises the potassium homeostasis, then perhaps we can engineer if we know the detailed biology or detailed physiology of these different components, then perhaps we can generate better, or we can do some genetic engineering, genome engineering, and then try to develop better crop plants. So if you look at the potassium transport element or potassium transport machinery, many of these potassium transporters are located at different level, like some of them are involved in the uptake of the potassium transport. This is a long distance transport of the potassium, and then the potassium is stored majorly in the leaf system in the vacuole. So there are many of these transporters which are distributed in these different tissues, different organs, and different cell types. And then they are regulated transcriptionally as well as post-transcriptional regulation is there. In post-transcriptional regulation, many of these transporters till now it has been identified they are regulated by phosphorylation, dephosphorylation mechanism. So there are protein kinases and protein phosphatases are involved in that. You know, we can simply see a kinase reaction, phosphorylation reaction, and a phosphatase, which is basically acting as a switching back the phosphorylation, dephosphorylation act as a switch. Now, I think one of the important and primary, the signaling pathway identified in the reported in the plant system for regulating the potassium transport or potassium signaling and transport mechanism is mediated by calcium signaling, specifically comprising of this CBL and CIPK. So these CBLs are what? These are called calcineurin B-like protein, because in animal system we have calcineurin A, which is a phosphatase, and it has a calcineurin B-like component or protein, which is a calcium sensor. And when there is an increase in the calcium concentration upon a signal transduction event, this senses the calcium, or they bind to the calcium and get activated, and this actually is a phosphatase. But in the case of the plant, instead of a calcineurin, we have another system, or there is a different kind of a component which is called CBL, which is the reminiscent of the CNB, and this phosphatase is actually a canonical phosphatase is missing, and instead of this phosphatase, this pair up with the kinase, and this form a module, and this perform the similar kind of a function, like when there is a transient rise in the calcium concentration upon a stress kind of event or a signaling event, it senses it and then get activated, and then this module regulate a large number of processes. And in Arabidopsis now we know, and even in different crop plants also we know now that these CBL and CIPK regulate a large number of physiological and biological processes. So there are many of these, reports, and then some of them I have, I'm highlighting here. This shows that this CBL and CIPK, which is a calcium signaling module, regulate the potassium uptake and signaling in Arabidopsis. So if you look at the component of the CBL and CIPK, there is a very important component called CIPK23, which is also known as a SOS2, because this is the same kind of a component which is identified as a salt overly sensitive by Professor Jiang and Zhu group. So this is involved in the salt tolerance as well. No, so this is the similar molecule. SOS2 is actually CIPK24. This is the similar kind of a molecule to the SOS2. So this kinase actually perform a very important kind of a function. So by genetic or by mutant-based approach, it has been identified that if you grow the plants, the mutant plants and then the wild-type plants in the normal media, they grow normally. Now, if you reduce the concentration of the potassium in the media, you see the growth of the overall growth of the plants or overall growth of the seedling is drastically inhibited, as you see, as you are approaching toward the very, very low concentration of the potassium. But when you see these phenotype of these mutant, they are super sensitive to this. Similarly, there is another kind of a kinase identified from the same family of CIPKs, which belongs to the SOS2 or CIPK24 family. This is called CIPK9, which also show the similar kind of a phenotype, right? And then there is another phosphatase identified, which this phosphatase interact with this kinase CIPK9. And when you mutate this phosphatase, the plants are able to grow better than the wild type, much better than the mutant. So they become tolerant. So when you mutagenize or when you make a mutant of this CIPK23 and CIPK9, the plants are super sensitive to grow under the low potassium media. But when you make the mutant of the phosphatase, then the plant becomes tolerant. So based on this, I think there is a lot of information, a lot of data is available. I think we can, in nutshell, we can briefly summarize this the functioning of the calcium-mediated signaling pathway, which regulate the potassium transporters in Arabidopsis through phosphorylation, dephosphorylation switch. This is the typical CBL CIPK, which is regulating this is the high affinity potassium transporter called AKT1. So this CBL-CIPK, specifically CIPK23, bind with this AKT1, phosphorylate it, and then upon phosphorylating, this become active, and then this, can take more and more potassium into from the soil. At the same time, there is a phosphatase which keep dephosphorylating it. So this is a phosphorylation dephosphorylation switch. When the plants are not growing when the plants do not need, the potassium, or they are not growing in the potassium deficient condition, this is basically saving the energy. They continuously dephosphorylating the channel, and then it is actually keeping the channel in an active state. Similarly, we have found out that there are many of these interactor of these kinases. This kinase called CIPK9 belongs to the potassium transport family. This is the HEC5. This is a transporter. There is another transporter called CUP3 and TPK4. This is the channel. So these are actually, this kinase is interacting with these transporters and channel and regulate these. So this is the information from the Arabidopsis. So if you summarize the knowledge from the Arabidopsis model system, we can summarize that there are briefly, this is the important kinase called CIPK23, which regulate the transporter AKT1, phosphorylate it, and at the same time there is a negative regulator, phosphatase, which keep dephosphorylating the channel and keeping the channel inactivating. So there is a phosphorylation dephosphorylation switch. At the same time, there is another kinase which is called CIPK9, which phosphorylate or which regulate this HEC5, TPK4 and CUP3. And there is another phosphatase like this AIP1. There is another phosphatase called AP2C, which I show you the phenotype for the mutant. And this keep dephosphorylating the kinase and then basically negatively regulating this pathway. So there are two different pathways simultaneously going on and regulating the potassium signaling or response pathway. So based on this information, now I think in Arabidopsis we know about these kind of the knowledge. The knowledge is there. So what we can do specifically in the case of the rice, and as we know that the rice require a lot of application, a lot of this NPK based or potassium based, and it is actually sensitive to grow in potassium deficient soil. So can we improve this the rice crop to grow, so that we can save the one important aspect would be if we can basically generate this tolerant plant which can grow in a low potassium or potassium deficient condition, then we can save the application of the potassium based fertilizer. And then this will also be environmentally friendly. We are using less of the chemical fertilizer application, and more economical to the farmers. So this cartoon represent the strategy. This is the plant growing in the potassium sufficient condition, and when they are growing in the deficient condition, they basically show the developmental phenotype. They are not able to grow. You see the drastic reduce in the crop productivity and yield. But somehow you can engineer these components specifically targeting from the calcium signaling and potassium transport component. Can we generate this kind of a plant which can grow in the potassium deficient media without any yield penalty or without any kind of a crop loss? So taking this model, I think I wanted to emphasize on that now we have pinpoint, or now we have actually found out there are many crucial component which can be targeted for genetic engineering. One is a CIPK23, another is a CIPK9, and these two phosphatases which are negative regulator, these two are positive regulator, and these channels and transporters which are the positive regulator. So we can utilize actually the genome engineering tools to generate the loss of function of these phosphatase, which are the negative regulator, and at the same time we can basically do a gain of function of these kinases as well as the transporter to generate the gain of function. So this is another cartoon or slide which depict the similar kind of thing, and here we are targeting the CRISPR-Cas based genome editing. In this case, what we are what we have done is you have basically generated the CRISPR-Cas line for these phosphatases, which are the negative regulator, and we are generating the gain of function by constitutively activating this positive regulator like kinases and then the transporter, which I'll be mentioning later. And when we can do this later on, we can, when all these individual lines can be then later on can be cross, and then all these can be pyramid together, and ultimately we can generate those kind of the kind of a variety which can be better tolerant to grow in the potassium deficient soil. So specifically for generating the gain of function for this positive regulator, I think we are doing precise base editing mechanism. Here what we are using is, this is the phenomena with the kinases. In the kinases, there is a very important reason which is called activation loop or activation segment, and in this there is a threonine or a serine which require phosphorylation, prior phosphorylation to get activated, and when this get phosphorylated, this become activated. So most of the signaling pathway, upstream signaling pathway, activate this phosphorylation or autophosphorylation of this residue. So in this case, this is a already reported knowledge in the biological system, that if you can create a phosphomimic version of this variant, convert the serine or threonine to aspartate, this will mimic, the aspartate will mimic like phosphoserine or phosphothreonine. So if you can convert the threonine or the serine into aspartate, this actually act as a mimicking for the phosphorylation mimic. So now it does not require, or you can bypass the upstream signaling pathway, and this become constitutively active. At the same time, now I think in these transporters and channel, we have identified the, through the in silico analysis and other analysis, we have identified that there are many potential serine and threonine site which can be phosphorylated by these kinases. And then these can be the target where we can, convert this threonine and serine into aspartate and make constitutively active, or we can say phosphomimic variant. So now in nutshell, we are handling actually two, as I mentioned, the kinases, the kinase 9 and 23, which are the positive regulator, and then the two protein phosphatases, which are the negative regulator. One is the AIP1 and AP2C1. And then there are the four transporters. Two of them are transporter, two of them are the channels, which are also positive regulator. And we wanted to create the phosphomimic of this kinase as well as the phosphomimic of these transporters, so that we can generate the gain of function. So before that, what we have done is, we already know that this the kinase 9, CIPK9 and 23 interact with these, in the Arabidopsis system. Based on this information, we have, I think we translate this knowledge into the rice, and then we found that, that these transporters interact with these kinase, the like HEC5 interact with the kinase, CIPK9, and then TPK4 interact with the kinase 9, and then the CIPK23 interact with the AKT1. So this has been also validated in the rice system. So now, as I mentioned that, you see, we have these potential the phosphorylation sites in the transporters. So these transporters, we wanted to actually pinpoint which one is the real the site for the phosphorylation by the kinases based. So we have used yeast complementation based approach, where what we have done is we have used this double mutant of the yeast called TRK1 and TRK2. If in the yeast system there are two transporters, TRK1 and TRK2, if you mutagenize this, the yeast is not able to grow in the low potassium media. As you see here, it is able to grow in the high potassium media, as you can see here, right? And if you grow them in the low potassium media, this is the mutant, right? This is the wild type yeast. It's not able to grow. But when we complement this, now I think this is our kind of a working model, which to identify which one of the phosphomimicking variant is active, or which one we should take further for the genetic engineering approach. So we have basically generated site-directed mutagenesis of all these variants, as I mentioned here. So all of them has been generated by site-directed mutagenesis, converting the either threonine to aspartate or serine to aspartate. and then transform these into the yeast system, and then found out that some of them, not all of them, are active. Some of them are showing the potential activity, and then they can be the potential target for the phosphorylation. So those are the candidates we have utilized, or we are using for generating the precise genome editing approach. So this is the brief slide where we have generated the loss of function mutant for the AP2C and AIP1, which are the negative regulator by the conventional CRISPR-Cas methodology, and this is the another, and the approach. This is the approach we are using for generating the converting these kinases, threonine to aspartate or serine to aspartate by HDR or homology directed repair. So in this case, we are not using a conventional kind of a CRISPR-Cas method, where here we are providing a kind of a template DNA repair template, including this mutation, the either threonine to aspartate or serine to aspartate. And then in this modified version of this homology directed repair, we have generated the variants of CIPK9 and CIPK23, and we are on the way of generating for these transporters. So we have done the transformation of rice with the CRISPR-Cas constructs and generated the plant. We have done the screening and identified, through the sequencing, and then identified these many of the line which are showing the the loss of function, as well as the conversion from threonine to aspartate or serine to aspartate in the phosphomimic virgin variant. So after that, once we generated these lines, we have generated many of these lines, and we have done some preliminary analysis. So the preliminary analysis, because we are generating many of these lines and we are confirming them through the sequencing approach. Now, I think to take further these lines, we have developed this kind of a preliminary approach, which is used generally by using the leaf disc assay. So the leaf disc assay of these flag leaf of the rice has been generated, which are growing in the greenhouse. And then they were basically subjected on the media containing the potassium sufficient containing media, and then the potassium deficient containing media. You see many of these lines are showing, kind of chlorosis and bleaching. So based on this approach, we have confirmed now at least some of the line for the kinase CIPK9. So this is the phosphomimic version. So these are the three lines of the phosphomimic version of the CIPK9, as you see here. And when they are floated, or when they are the leaf disc are floated on the potassium sufficient media, you see there is hardly any loss of the chlorophyll, and this is the wild type. When you decrease the concentration of the potassium in the media, specifically you reduce or you omit completely the potassium from the media, the wild type is performing very poorly and losing the chlorophyll, which is the sign of the potassium deficiency. But these lines are able to retain to some extent the chlorophyll. So this is a potential kind of approach for screening. And although this is a preliminary kind of analysis, we will be doing all these analysis in the seedling, and then the mature plant stays in the greenhouse growth condition. Similarly, for the CIPK9 phosphomimic variant, we see the similar kind of a result. And when we have created the loss of function of these two phosphatases, the phosphatase AP2C and AIP1, as you see here, we are growing the plant in the hydroponic media. This is the potassium sufficient media, and this is the potassium deficient media. Now these plants are taken, the picture is taken, and as you see, this is if you grow the plant, if you grow the seeds directly on the potassium deficient media, you see how the growth of the seedling is arrested, drastically arresting the growth of the seedling in the wild type. But some of these negative regulators, these lines, we are screening more of these lines which are performing better than the wild type. So now I think based on this, we can now, based on this now this all is a kind of a preliminary data, but we are trying to approach toward doing more and more, confirmatory kind of experiments by once we get these planned, once we get the plant in the T2 and T3 generation. So then what I try to convey here is, based on this targeting the calcium signaling and then the potassium transport component, we have actually shown to some extent some proof of concept that if you downregulate or if we knock out the negative regulator like these phosphatases, we see a kind of a better growth in the potassium deficient media. If we make constitutively active or phosphomimic variants, and that will basically create a kind of a gain of function, we can also, impart the plant the tolerance to the potassium deficiency. So if we can combine these two or combine all these, in one plant, once all these traits can be combined in one plant, perhaps, we can generate a plant which is much tolerant to potassium deficient soil. And then, I think this will be very useful, specifically as I mentioned, we can reduce the application of this chemical based fertilizer, potash-based fertilizer, and which is heavily applied in the crop plants. So this is end of my presentation, and thank you very much. I wanted to acknowledge, most of the work is done by the postdoc Pooja Verma and then the GRF Sonam Ahuja. And this is the project in RICE is funded by the DBT, and my lab is also funded by DST, CSIR, UGC, BRNS, and Delhi University. And I am also thankful to other members of the lab whose work I have not presented here. Thank you very much for your kind attention.
Okay, thank you, Girder, for an excellent presentation, giving an overall beautiful account of the work you're doing. Right from function genomics to genome editing. I'm very happy to see the kind of progress being made in your lab. Thank you very much, and hope we'll have some questions at the end. And request you to kindly stay. Well, to move on, the another speaker from our national laboratories, one of them now, the last but not the least, for today's webinar is Dr. Viswanathan Chinnusamy. Dr. Viswanathan Chinnusamy is very well known in the area of abiotic stress tolerance, nationally, internationally. He has contributed significantly, particularly in the area of heat tolerance and cold tolerance as well. He was, I would say, the one with Dr. Jiakang Zhu to identify ICE1, in fact, the inducer of CBF expression, which was quite a useful transcription factor for the production of some of these improved abiotic stress tolerant transgenic plants. He's contributing through the process of functional genomics, genetic engineering, and now also, genome editing. And I'm very happy to also inform those who are not aware that Dr. Viswanathan Chinnusamy was the co-discoverer with Jiakang Zhu of the ABA receptor. Sometimes in 2009, the paper appeared in Nature, and a wonderful discovery, one of the top 10 discoveries considered in science, in the world. So we are very proud to have Dr. Viswanathan on the panel and making this presentation. He is heading the division of plant physiology at IRI, and also has created an excellent facility of phenomics, in the same laboratory and the institute, that is Indian Agricultural Research Institute, which was a seat of a green revolution in 60s, 70s, from Indian perspective. So I therefore have the privilege to invite and welcome Dr. Viswanathan for making his presentation. Dr. Viswanathan, the screen is yours.
So today I'll be talking about genetic engineering and genome editing for enhancing abiotic stress tolerance of crop. So I'll just skip this introduction, why we have to improve abiotic stress tolerance. And we have chosen the crop rice because rice is one of the 4 crops that contribute to the 2/3 of the global agriculture calories. And India, if you see, rice is contributing to about 40% of the agriculture food production. So rice is very important crop, and rice is also sensitive to various abiotic stresses, and water is one of the key input for rice. And if you see the global map, which is shown here, in India, if you see, India is under water scarce line by 2050. So we have to produce more rice and from less water, and which is a greater challenge. Besides the water requirement, the salinity is increasing, and high temperature is also increasing, another problem. So to address all these problems, various approaches are being used, and my previous speakers have elaborated various approaches. Our lab has taken up two different approaches, that is genetic engineering and genome editing for various abiotic stress tolerance in rice. So I will be presenting some of the work which we have done recently on genetic engineering by using ABA receptor and the inducer of CBS expression 1, and genome editing of drought and salt tolerant genes in rice. So as Professor Bansal mentioned, abscisic acid was discovered in 1960s, but the receptor was discovered only in 2009. It took almost about 50 years. So since abscisic acid controls various traits from stomatal control, which makes the free water loss from the leaf into the atmosphere, which almost uses about 99% of the water which is taken up by the plant. So which is a very key process. Tight control of stomata is very important to save any water in agriculture. But at the same time, it is also promoting root growth under drought stress condition. It provides extreme stress tolerance to various stresses, not only to drought, but also to other stresses. And the hormone, although it is known as plant stress hormone, it is also important for the plant development. And if ABA is deficient, as you can see from this picture, the plants will not grow normally. And if ABA is deficient or if signaling pathway is impaired, the seeds germinate on the mother plant, and dormancy is lost, and there is a problem in the seed development also. Besides, ABA also play a very important role in the biotic stress responses, mainly in case of necrotrophic pathogens. ABA provide tolerance, that is positively regulates, whereas in biotropic pathogens it can negatively regulate. And for some of the insects, it positively regulate tolerance through zasmonic acid signaling. Besides the plant, ABA is also very important for human beings, and since it regulates the innate immunity as well as the pancreatic insulin release, and it also plays a role in inhibition of cancer cells. So ABA is a pro-survival factor in human being also. And the ABA receptor discovery has clearly showed that what are the various components are there, and how it works under non-stress condition and stress condition, and then how these stress responsive genes are expressing, and how the stomata is closed. So in the absence of stress condition, the negative regulator of ABA signaling, that is protein phosphatase 2C, it interact with SNRK kinases, both SNRK1 family as well as SNRK2 family, and it dephosphorylates the SNRK kinases. So once the SNRK are dephosphorylated, the downstream targets are not phosphorylated. So there is a energy signaling as well as stress responsive gene expression is not occurring. In the presence of stress, when the ABA content increases, ABA binds to the receptor, that is the pyrobactin resistance 1 and pyrobactin resistant 1 like receptor, which are 14 in Arabidopsis. So the ABA binds to the receptor. Binding of ABA causes conformation change, which help in sequestration of protein phosphatase 2C. That is, the ABA bound form of ABA receptor can bind to the protein phosphatase 2C. Now the SNRK2 kinases are free from the repression. And then these SNRK kinases, they can phosphorylate the downstream target, and the SNRK1 is involved in the energy signaling, and then it regulates the carbohydrate metabolism and growth and adaptation. SNRK2 kinases, they play a very important role in stress responsive gene expression. As it is shown here, it can phosphorylate various transcription factor, and then it regulate gene expression, or it can also regulate various membrane protein, such as the potassium channel, which is present on the stomatal God cell plasma membrane, and the slow release anion channel, and the respiratory burst oxidase, and the aquaporin. So various ion channel proteins are phosphorylated. This phosphorylation either positively or negatively regulates this ion channel protein, and thus it leads to stomatal closure. So it plays very diverse role from the development to stress response. So once the ABA receptors were discovered, there are different approaches have been taken for using it for enhancing the drought tolerance and water use efficiency in different crops. So one of the simple approaches overexpression of ABA receptor by using transgenic approach. The other two alternate approaches are to produce an orthogonal receptor that can be activated by off-the-shelf agriculture chemicals and identification of cheap agonists to ABA receptor. So on some of these aspects, I worked when I was in Professor Zhang Hang Zhu lab. So currently, our lab is mainly focusing on the overexpression of ABA receptor. So I will be presenting some of the results from these studies. So if you see the hormone receptor report here, if you see for the other, all the classical hormone, abscisic acid receptor has the maximum number of hormones. That is, it has about 14 receptors in Arabidopsis. In rice also, we have identified about 13 receptors. Of the 13 receptors, 2 are non-functional, so at least 11 are functional receptors. So to understand whether they have a redundant role or non-redundant role, why the plant should have so many receptors, especially for ABA. So we analyzed the expression pattern to begin with in different tissues, and as seen from the real-time RT-PCR expression analysis, you can see that some of the receptors, they are highly expressed in the seeds as compared to the base level expression in the root in seedlings. So these pile 8 and pile 9, you can see highest expression in the seed. Some of the receptor, they express so highest expression in the panicle. Some they show highest expression in the stem and flag leaf. So depending upon the receptor, they expression varies in different tissues. And we also examined the stress responsive expression of all these receptor, and you can see from this graph that is in some of the tissues these receptors are upregulated, and it can be upregulated both by drought and heat, or it can be upregulated either by a drought. In some cases, it can also be downregulated. So it is a kind of differential regulation is observed. So this suggests that there is a non-redundant function, and it may have different roles and different kind of stresses. So to understand the functions of this ABA receptor, we developed transgenics either by using the RD29A promoter or by using the ubiquitin promoter, and also we attempted the RNAi approach. So these are, I will be presenting results for 2-3 receptors which have some contrasting results. So this is the results for the one of the ABA receptors. So when we developed the pile 6 transgenic plant and confirmed by southern blot analysis and also by qPCR analysis, and to begin with, we examined the ligand concentration, that is abscisic acid is the pan agonist for all the receptors. So we examined whether any change in the ABA content is there. And as you can see from this quantification result, the abscisic acid content was significantly higher in this overexpression plant. And if we see the rate limiting enzyme, that is NC9-cis epoxycarotenoid dioxinase, so it has different family members, and most of the family members were upregulated in the pile 6 overexpression line. So this happened not only in case of ubiquitin overexpression, in case of RD29A expression, as well as in RNAi line also we found this. So this suggests that a specific level of pile 6 is important for the abscisic acid homeostasis. Then we also examined root rate in the lab condition. So it enhances the root rate, that is overexpression enhances the total root length as compared with the wild type or the RNAi lines. Then we also examined the dehydration tolerance in the pot culture condition, and these transgenic plant which expresses ABA receptor, they have better survival rate as compared with RNAi line or the wild type plant. And this better survival was due to enhanced membrane stability or reduced the ion leakage, and also expression of various stress responsive genes that contribute to dehydration stress tolerance. And in the transgenic plant overexpression line, these genes expression was significantly upregulated as compared with the wild type plant. In case of RNAi plants, some of these genes expression were significantly lower as compared with that of the wild type plant. We also examined the water loss by using the excise leaf water loss assay. As compared with the wild type and RNAi line, the transgenic lines used less amount of water, and the scanning electron microscopy also showed that as compared with the wild type plant, the transgenic plant has relatively closed stomata. And we examined the whole plant water use, and as you see from these two graphs, the amount of water which is used by the wild type plant in 30 days is sufficient for the transgenic plant for about 45 to 50 days. So in order to examine whether these differences is due to the difference in the biomass or leaf area, we converted the water use per unit leaf area, and as you can see from this particular graph, so it's uses about 22 to 27% less water as compared with that of the wild type. In the adult plant stage, also under the transgenic greenhouse condition, it produces very high stress tolerance. That is, once these plants are exposed to stress, and then when they recovered, only the transgenic plant can survive better as compared with the wild type plants. However, if we see the yield level, the pile 6 overexpression transgenic plant produce less biomass, and also it produce less yield under normal stress condition, which is not desirable. So overexpression resulted in the reduction in the plant height, and also a reduction in various yield component rate, including the number of grains. So precise regulation of this receptor restricted to the vegetative stage may help enhance the dehydration stress tolerance without affecting the yield. The other receptors we have analyzed was the pile 10, and the pile 10 belongs to the subfamily 1, whereas the pile 6 belongs to the subfamily 2 of the AB receptor. Again, the similar kind of analysis was carried out. So the pile 6 did not provide tolerance to cold stress, but this pile 10 transgenic, it provide a high level of tolerance to cold stress, and wild type plant could not survive under these stress conditions, whereas the transgenic plants they showed better survival. And the better survival was associated with some of the downstream transcription factor as well as the effector genes, as we can see from here, that is core 410 or dehydrin or LEA3. These genes express significantly higher level in this transgenic plant. We also examined the tolerance level at adult plant stage. So in the photosynthesis rate, it was slightly higher photosynthesis rate was there under the control condition. And both the wild type plant and the transgenic plant, they were grown in the same pot and exposed to the stress. And under this condition, in the drought condition, there was no conclusive results for the photosynthesis. However, water use was significantly low for the transgenic plant as compared with that of the wild type plant, as it is shown in this graph. And water use efficiency was better for the transgenic plant, both in the control condition and under drought stress condition for some of the event. And the grain yield was under control condition, it was compromised, but under stress condition, these 3 of these even produced higher grain yield as compared with that of the wild type plant. And this transgenic plants also expresses some of the effector genes for dehydration stress tolerance, such as RAP16, LEA3, and various other genes at a very high level as compared to that of the wild type plant. Similarly, we have analyzed other family members also. This is subfamily 1, so I have given one example for each family. So this subfamily 1 receptors were examined for their osmotic stress tolerance, salt tolerance, and cold tolerance. So these receptors at seedling stage, so this is comparison of constitutively expressing transgenics, and this is transgenic plant that expresses file 1 and from the RD29A promoter. So under PEG stress condition, the transgenic plant showed better survival as compared with the wild type plant. And this is salt stress treatment at adult plant stage. So when we give salt stress, most of the like wild type plant, they could not survive at adult plant stage, whereas the transgenic plant survived. And this was also reflected in the sodium potassium ratio as compared with the wild type plant, which the sodium potassium ratio was high, which means the plant took more sodium as compared with potassium, whereas these transgenic lines, they took less sodium. And in the leaf disc assay also, these transgenic plants performed better. So these are pile 1 appears to be more promising for producing lines with enhanced salt tolerance. And again, for PEG stress tolerance also, as it showed at seedling stage, it can tolerate PEG stress better. And also under adult plant stage, when we subject them to stress, it produced better survival rate. And these transgenic plants produced more robust root system as compared with that of the wild type plant. And these transgenic plants, pile 1 overexpression lines used less amount of water as compared to that of the wild type plant. So this is like similarly we have results for the other ABA receptors. So to conclude the ABA receptor story, in general, the subfamily 2 receptors, when we overexpressed, we found that enhanced spikelet sterility, although it can provide extreme tolerance to various kind of stresses. The subfamily 1 and subfamily 3 appears to be better candidates for producing transgenic without compromising the yield. However, precise manipulation of these receptors is very important. Constitutive overexpression or even the stress-inducible overexpression at reproductive stage may lead to reduction in the yield loss. And we also engineered the rice plant with the ICE1 gene, which is playing a role in cold tolerance. And also from in several crops, it has been shown that overexpression lead to multiple stress tolerance. So we examined this story in the rice. So the overexpression lines were analyzed for the target genes such as the transcription factors and various effector genes involved in the triglyceride synthesis and various other genes. And these transgenic plants expressed high amount of the downstream targets. And then we analyzed tolerance to various stresses. This is the cold stress response of the wild type plant. The recovery is very poor, whereas the transgenic plants, they showed very good recovery and they showed better stress tolerance at seedling stage. And this is again seed tolerance at the adult plant stage. So these plants performed better after under the cold stress in terms of the chlorophyll fluorescence as well as in terms of grain yield after recovery. And we also examined tolerance to salt stress, and again in the salt stress also these transgenic plants showed better spikelet fertility, that is sterility is less, and it also showed better grain yield under salt stress as compared to that of the wild type plant. And the similar results were obtained for the drought also. So these overexpression lines produced higher grain yield both under control as well as under the drought stress condition. So this is the yield under normal condition, unlike the AB receptors case where the yield under normal stress is compromised. In this overexpression line, yield under normal condition is not affected, and it also gives multiple stress tolerance. So the last part of my talk is on genome editing. As we have seen that this year Nobel Prize was awarded to the genome editing discovery to Jennifer Doudna and Emmanuel Carpentier. So we used the genome editing approach, explored genome editing approach to improve drought tolerance in rice. So to us, this is simple to show how genome editing works. So it is the people, those who are working with transgenic will be very familiar. So similar to the transgenic, here also we make gene construct, and which is having two different expression cassette. One is the guide RNA expression cassette, and another one is Cas9 expression cassette. So this tDNA is transformed into rice, and transgenic plant is produced. Once the tDNA goes into the nucleus and it integrates into the genome, it produces the CRISPR RNA, that is the guide RNA with scaffold RNA and the Cas9 nucleus. And these complex look for its complementary region in the genome, and then the Cas9 endonuclease makes a double-stranded cut in the genome. And this double-stranded cut, when it is repaired, it is mutations are produced. And one of the advantage of Cas9, CRISPR Cas9 technology is that once it is transformed and the transgene goes and integrate into one place, and the target loci is in different place. So once the mutation is created, these two loci can be easily separated. So you can separate the tDNA containing plant and the plant which is having the mutation. So you get a plant which is free from tDNA, and the mutant is indistinguishable from that of the mutant that you produce by conventional approach. So once the double-stranded break is created on the genome of the plant, this double-stranded break is repaired by the non-homologous end-joining repair, which results in the mutation, or it can be provided with a template as shown by Dr. Girder Pandey. So we can produce a specific mutation in this, or you can also place your transgene in a safe harbor loci. So these are the various approaches are being used. So we selected one of the genes which is known as drought and salt tolerance gene, which was identified in 2009 by screening 270,000 EMS mutant, and this gene encodes for Z2H to zinc finger transcription factor. When there is a loss of function mutation in this particular zinc finger domain of this transcription factor, these mutant plants are more tolerant to osmotic stress as well as sodium chloride stress. And we also try to identify this mutation from the Nagina 22 mutant population, which was generated by Professor Mahapatara's lab. However, the similar mutant was not identified, although one mutant was identified which was tolerant to PEG stress. This mutant had some adverse low yield or the grain quality problem, so hence and the gene was also could not be mapped so far. So it is not easy to get a specific mutation even by using the conventional EMS mutagenesis, because it is a random process and it is difficult to get the mutation. So use of this Cas9 technology will be very useful. So we have designed two guide RNAs for the DST gene. These are the genes, the guide RNAs which are shown in the bold letters. And then these two guide RNAs were cloned in the CRISPR-Cas9 vector. That is, two guide RNA were put in one vector and the plants were transformed. And then in the T0 generation, the DNA sequence was done for the DST gene, and we could identify many of the mutation. And this DNA sequence in the T0 generation, often it is in the heterozygous stage, so mostly we difficult to get homozygous mutants. So these mutants were analyzed by using various softwares, and we could identify a large number of mutants. And we selected one of the mutant for detailed analysis here, which is shown here. So this mutation creates 360 base pair deletion. So here from this guide RNA and between these two guide RNA, whatever the sequence is there, which is deleted because of the double-stranded break between these two regions, and which can be easily confirmed by PCR. And in the T3 generation, we have also identified plants which do not have this tDNA and they are hygromycin sensitive. And we have selected those plants for the phenotyping. And these mutant plants, as expected, which behaved similar to that of the EMS mutant, which was identified in 2009. This has a broader leaf. And we also examined the water use by this plant, because DST gene is known to regulate stomatal control through the regulation of the amount of hydrogen peroxide that is accumulating inside the stomata. This we examined, and we found that this transgenic plants, the genome edited plant, they use less water as compared to that of the wild type plant. And we examined, besides this stomatal closure, whether it has any role in the stomatal number. Interestingly, we found that this gene also regulates the stomatal number. If you see this graph, it has the DST mutant has about 17% decrease in the stomatal density. Then we examined various genes involved in the stomatal development in the developing leaves of the DST mutant, and we found that three of the genes, that is Teach1, mute, and ICE1, these genes expression was significantly reduced in the developing leaf. And we also found that these promoter of these genes have the DST binding site. So further, these need to be validated whether it really binding to these genes and regulate the stomatal development. And we have also examined the tolerance of this mutant in the seedling stage, as examined in the 2009 paper. So it showed that for PEG as well as for NaCl, it shows a tolerance, and the survival rate, there is significant difference in the survival rate of this DST mutant. So further, these plants are being analyzed at the adult plant stage. So besides this allele, we have also developed other alleles, and various alleles are being examined for their stress tolerance now. So we hope to get some mutants which produces better stress tolerance from this genome editor line. So, to summarize, the ABA receptors have a non-redundant role in stress response and plant development, and ABA receptor engineering can enhance water use efficiency and tolerant to multiple stresses depending upon the receptor which we use. However, tissue-specific as well as stress-regulated expression is the key to use this receptor for enhancing the stress tolerance. ATIS1 can be used to enhance grain yield in rice under non-stress condition as well as in the various stress condition. CRISPR-Cas9 mutagenesis is a useful tool for creating stress-tolerant plant. And testing of this developed genome edited or genetic engineered lines under field condition is necessary to develop really stress tolerant product, because currently all these plants are being examined only under greenhouse condition. Hope we will soon be able to do it under field condition. So with that, I thank the funding agencies, that is NISF, ICR, and DBT Government of India, and ICR, IARI. And I also thank the students, those who have done this work. Santosh Kumar, who has done work on the genome editing as well as ABA receptor work, and Shashank has done ABA receptor transgenic. Again, Rakesh, he worked on the ICE1 transgenic, and all the other students, they have contributed. And I also thank Professor Zhang Kangsu for providing various construct for genome editing. So finally, I thank Professor K.C. Bansal and Dr. Ajay and Dr. Himanshu Pathak for providing me this opportunity. So one who solves the problem of water is worth 2 Nobel Prizes, one for science and one for peace. So every one of us should try to conserve water and make effort to reduce freshwater use. Thank you.
Sure. Thank you so much. We hope it soon happens that we get 2 Nobel Prizes anywhere in the world who is working on these. Well, thank you so much, Dr. Vishwanathan, for this excellent presentation, and congratulations for developing some of these transgenic and genome-edited rice crops with various useful genes. Well, sorry for the delay. We have 15 more minutes now. Before 8:30, we must close. Before I thank all the speakers profusely again, I thought we would take up a few questions. And of course, I'm very thankful to the presenters, particularly Professor Mark Tester and Dr. Hima Bindu, for taking up these questions and answering them in the chat box. Nevertheless, I think few more questions we will take up. before I request Dr. Himanshu Pathak to conclude. So I think to start with, I will have a question myself, which I don't think there is any question which is unanswered in the chat box, but I have one to Professor Mark Tester, and that is, Professor Mark Tester, I'm sure you're there. Sorry for keeping you, busy for so late. Professor Mark, are you there? Yeah, thank you, Professor Mark. The question was that while it's a very good idea to domesticate some of these wild crops and make them as new crops feasible, possibly. And so is that also possible that while working on domesticating some of these wild crops that we deal with some of the negative regulators of yield, in those crops, that we do have some advantage, rather they thrive in some of these, suboptimal conditions, but will we have some advantage of having little more yield as well, not as high yield as any other elite genotype would have it, but will they have some advantage for yield as well, apart from their, withstanding the various suboptimal conditions?
I don't know, to be honest. I think that in the real agronomy and the real fields, that some of these plants could well perform very well, because what we're starting to see is that they not only have, some of these plants have not only very good resistances to the abiotic stresses like salinity, but also have very good biotic stress tolerances as well. And I mean, these biotic stress tolerances are famously, this has been done for decades, bringing in biotic stress tolerance from wild relatives. And this is done from grafted tomato rootstocks, all the way through, of course, to cereals and integration of resistance alleles from wild relatives. So I think that could lead to some significant increases in yield. I don't know about negative regulators per se, to be honest. I'd be a little bit surprised, to be honest, if there's significant, raw total yield potential benefits from these wild relatives. I think it's primarily beneficial for these abiotic and biotic stress tolerances.
Sure, thank you. In fact, there was another similar question by Dr. Monica. She has already left, actually, from Delhi University. The question, of course, you've already taken it up, and but for the sake of the clarity for the audience, that yes, these neglected crops, no doubt, are very important, and they are low yielding. But I think nutritionally they are quite rich. So therefore, we must try to, in fact, value them for the nutrient content rather than yield alone. As we normally do with starchy crops, whether it's wheat or rice or maize.
That's an excellent comment. That's an excellent comment. And yes, we have to, I think all of us need to consider not just the delivery of more and more calories more sustainably, but also the delivery of high nutritional value, the micronutrients in particular, very, very important. That's a very good comment.
Well, yes, for this immunity, particularly as we're going through this damn pandemic, so.
Well, this is terrible, of course, this pandemic, but it is increasing, I think, the world's, understanding, appreciation of the work that all of us do, which is only good.
Great. So thank you very much, Professor Mark Tester. I don't see any other question for you here in the chat box, and again, profusely from all of us from India, and I'm sure again from the participants, we profusely thank you, for this excellent presentation.
Thank you very much. Thank you very much. If you want me to stay around for 10 more minutes, I can, but I have to go sharp.
I know you have got another appointment, but if there are some questions, certainly, but otherwise, once again, thanks to you.
It's a pleasure to help. Thank you so much.
Thank you so much, Mark. Thank you for honoring our invitation. Thank you very much. Yeah. So we move on, and I have one more question, actually speaking, to Professor Girder Pandey, and this is from the convener himself from this institute, Dr. Ajay Singh. And question, Dr. Pandey, is there any role of this potassium signaling in regulating the transcription factors? And as a result, if you can target those transcription factors, can we try to have multi-stress tolerance as well, rather than only?
Yes, a very interesting question. Till now, nothing is known, because all this in plant system is mostly regulated through post-translational phosphorylation and dephosphorylation of which. But we don't know, and in bacteria, we know, I think there are many transcription factors which regulate this pathway. But in plant, we do not know exactly what kind of a transcription factor is involved in the potassium response, potassium signaling pathway. So it would be very interesting, and I think in fact many people are trying to find out this, but still we don't know much. But hopefully we have something.
Okay. Thank you. Well, there's another question just dropped in, and I think Dr. Vishwanathan has already tried to answer it, but still, I will take it up that whether, we can have a single gene or can we tackle together both, drought and salt stress, using the kind of approach which you're using, particularly, let's say, targeting DST gene. This is from Dr. Rajgopal from Germany.
Yeah, thank you, sir. I think we have to see, because the DST gene currently we have examined only at seedling stage stress tolerance. So we have to see if it can give adult plant tolerance to salt and drought stress separately. Combining these two stress, we have not attempted even for any of the transgenics we have developed so far. I think it will be too much to expect from a single gene to provide tolerance to these stresses. Even if it provides tolerance to one stress also, that itself is I think we have to work with multiple genes, not a single genes may not be providing tolerance to that extent.
But for 2 Nobel Prizes, we need, I believe, a single gene anyway.
Yes.
Okay, so, well, there's another question, probably that could be the last question to Dr. Pandey, Girder Pandey, is there any crosstalk in case of abiotic stress between potassium and phosphorus?
Yes.
This is the question from...
Does potassium talk with the phosphorus as well as nitrate and calcium, obviously. So this has been shown, many physiological experiment has been shown. And genetically, we don't know exactly what are the components, but based on the physiological experiment, it has been shown that it crosstalk with the phosphate and nitrate, obviously nitrate. So nitrate, we know a lot of crosstalk through genetic component, because some of these, as I mentioned, some of these kinases and then sensor calcium signaling pathways are commonly shared by the nitrate sensing mechanism as well as the potassium sensing and response pathway. But phosphate, yes, I think we don't know. But when we do many of these transcriptome profiling, and we find in these mutants and under the potassium deficiency condition, so we find many of these phosphate homeostasis related genes are either upregulated or downregulated. So it may have definitely some kind of a connection with that.
Okay, thank you, Dr. Pandey. And this is the last question, and that is actually from Dr. Somdat CPR Shimla. I couldn't ignore this question because it's a little different, that he's asking whether genome-edited plants need to undergo biosafety guidelines. And I can try to answer him briefly on this. I know Dr. Vishwanathan or Dr. Pandey would also agree with me, that yes, they need to undergo. The direct answer is they need to undergo the biosafety regulatory guidelines. In India, I've been talking about it. And these regulatory guidelines for genome-edited plants are still not out officially, though the draft has been prepared and has been submitted by Department of Biotechnology to the Ministry of Environment and Forest, and the apex body, that is Genetic Engineering Appraisal Committee, is looking at it at the moment. And as far as different types of genome editing is concerned, SDN1, because of we had a consultation with the National Academy of Agricultural Sciences, SDN1 type where there's no foreign DNA integration, and which is just again a mutagenesis like we had used to do random, but this is more targeted. We have requested that there should not be any kind of regulation, which is the case globally in many of the countries, including US. And there are genome-edited crops, which is through SDN1 category have been, already commercialized, for example, white mushroom in US. So I think we need to still wait and watch how we are going to develop our own guideline. Well, with this, I think we have run out of the time. And I also, on my own behalf and on behalf of the organizers, both the institutions and all the participants, take this opportunity to thank very profusely all the excellent speakers, and who very readily and kindly agreed to our requests. As you all know, Professor Mark Sester himself is such a busy man, such a busy scientist, and we are so proud that he is still around and available to this developing world, answering many of these questions and taking out time for his presentation. And also Dr. Matthew Reynolds, and equally, Dr. Girder Pandey and Dr. Vishnu Chinnusamy, and of course, Dr. Hima Bindu, from Professor Rajiv Varshney's lab from Microset, all doing excellent piece of work. And as I see from the chat box, everyone has liked the presentation, and kind of good information, good knowledge, ideas have been generated. And I believe this was certainly, the credit goes to all the presenters, and kudos and our, kind of cheers and thanks to all the presenters. Thank you very much. And thanks to all the audience, of course, and to those who asked several important, very good questions. And thanks, Mark, for staying that long for us again.
My pleasure. Absolute pleasure.
Yeah.
Best of luck to everybody.
Great. Thank you so much. You made it really a very important, webinar for all of us. Okay, and now I think I have 5 minutes more, and I request, Dr. Himanshu Pathak to kindly conclude. And sorry for a little delay in the whole process because of some technical issues.
Thank you, sir. Thank you very much. Thank you very much. I think I must thank you profusely, actually, because the whole show was thought, planned, moderated by you only. But friends, I think all of you will agree with me that we really had a very, very informative, very, very pleasant, and very, very useful, but of course, morning in some other countries. And as Dr. Bansal started saying that India needs this technology because Indian agriculture is facing all kinds of abiotic stresses. And as I also fully agree with when Dr. Mark Testa said that we need sensible, sustainable solutions. We really need sensible, sustainable solutions for abiotic stress management. Friends, as we have seen today evening that we started with salinity, went to drought, nutrient deficiency, heat, cold. I think almost all the abiotic stresses we have covered. And crop-wise, we didn't leave really anything. We talked about rice, we talked about wheat, chickpea, and of course, neglected crops. But I don't want to tell them as neglected crops. Those crops are now going to become very, very important crops. Very, very important crops from climate resilience point of view, from abiotic stress point of view, and so on. And probably the lesson which we could learn today, that there are several new developments, very, very, very useful emerging developments are taking place. And the future is really bright. But we have to really go step by step, step by step. And as I think Vishwanathan said, maybe a little jokingly, that we should not expect too much from one gene. So probably we should not expect too much too quickly from all the developments which are happening. But of course, developments are in sight. Science is progressing, starting from plant designing to CRISPR-Cas9, genome editing, multi-omics, and all of these things. But I think at the end of the day, we all should realize that we certainly need networking. Networking with all the scientists who are working in this particular area, not only within our own country, but also outside. So these are the few messages, and I'm sure that all of us, like you see today, there are so many students, young researchers, and probably for them, it is really going to be very, very useful to know all these emerging areas of new science with such a brilliant presentation, with all these luminaries of these particular areas of research. Thank you very much, all the speakers. Thank you, Dr. Bansal, for your all efforts. I do not want to, again, mention all these things, starting from planning, moderation, and of course, our own Ajay, and everybody, all the audience from various universities, institutes of India and abroad. So I thank each and every one of them. Today, of course, it was online, virtual meeting, but certainly I would like to extend my own invitation from my institute, that whenever things become normal, we will be really very, very happy to have you here physically in our institute, and we shall look for collaboration, all your very, very important and useful work. Once again, Thank you, each and every one of you. Thank you very much. Take care, because COVID is still there with us. I'm sure it is going to go, but still it is there. So thank you very much, all of you. Now it is over to Ajay.
Dr. Ajay Singh for a brief vote of thanks.
Yeah.
Okay, thank you, sir. So, on behalf of organizing committee, I express my gratitude and heartfelt thanks to all the speakers, starting from Professor Mark Tester, Dr. Hima Bindu, Dr. Matthew Reynolds, Dr. Girder Pandey, and Dr. Vishnasan Chinusamy. It was a great pleasure to listening to you all. Their talks on different aspects of genomics for improvement of abiotic stress tolerance in crop plants were excellent and very informative. Thanks once again, sir, for sparing your time, your insight, and those discussions and deliberations. I humbly thanks to our Director, Professor Himanshu Pathak, for his continued support, guidance, inspiration, motivation to conduct this webinar. A special note of thanks to Professor K.C. Bansal for continued support and guidance to make this webinar a success. A big thanks to our technical team, Praveen More, Lalit Aher, without whom this webinar would not have been possible. Before I conclude, a big heartfelt thanks to all the participants from across the globe for their active participation. Really, their response during this webinar was very overwhelming. With this, we come up with closure of this webinar today. Thank you once again. Stay safe and have a nice weekend. Thank you, sir.